Okay, so good morning, everybody. And this is the third edition in the Highlighting Organic Chemistry Across Europe. And I'm very pleased that this third edition is going to be highlighting organic chemistry in Finland. So with that, I'll pass it over to Professor Ari Koskinen, who's the Finnish representative on our division. Ari, over to you.
Thank you, Patrick, and welcome from me too to everybody. I have to thank Juri Timonen for organizing the whole meeting, and I will just say a few words about the organizer. The organizer is the Finnish Society for Synthetic Chemistry, which was formed 30 years ago in Finland, although organic chemistry itself has been for much longer. But you will hear from that later on. Juri has put up a very good program. I think it's a nice cross-section of organic chemistry in Finland at the moment. The scene has changed quite a bit, and you will see there are quite a number of relatively new appointees. I will not take any further time here. I will just pass on the podium to the Juri, I think, who will start what organic chemistry is in Finland at the moment. Welcome from my part to everybody.
All right, I'll pick it up from here. So the very first thing we're going to look at is basically just looking at what's going on in Finland and where we are coming from. Can everybody see my screen?
Yes.
All right, perfect. So the idea here is that I've gathered kind of two distinct points here. The first point being, where do we come from in Finland in terms of organic chemistry? The second point is, where are we at now and where things headed? So the origins of organic chemistry in Finland really stem from the fact that we used to be a really undeveloped society, and we were kind of tossed back and forth between several other countries. But we've developed our own take on chemistry, and really the Finnish organic chemistry heavily relies on synthesis, on drawing inspiration and drawing kind of compounds from nature, as you can see. And then we also kind of get interested in biochemistry very early on. So I've gathered some key figures here that had an influential role on historical organic chemistry and kind of chemists that you probably should know from Finland. In the historical context, we have Gustav Kompa here. And Gustav Kompa was basically the first proper organic synthetic chemist in Finland. He used to work what is now Aalto University, where I'm working, and I'm actually currently the holder of his original professorship. And Gustav Kompa was hugely influential in Finnish organic chemistry. He was an extremely highly skilled organic synthetic chemist working on natural product total synthesis, but he also had a really keen eye on how to industrialize his discoveries. Everything had to be robust, scalable, and I have an example of what this means. So Gustav Kompa is one of the founding fathers of Finnish chemical industry and where Finnish organic chemistry actually is nowadays. Another key figure is Artturi Ilmari Virtanen, who's currently the only Nobel laureate in Finland who's received his Nobel Prize in 1945. Artturi Ilmari Virtanen originally was an organic chemist, and he carried out structural determinations of natural products. But then he kind of dwelled into biochemistry and agrochemistry, and actually he got his Nobel Prize for developing fodder preservants. His heritage is also extremely influential in Finland, especially the bioorganic scene heavily relies on his group and his original students. Then Lydia Seseman is the first Finnish female to ever be granted a PhD, which is as early as 1874, and she was also an organic synthetic chemist. She was doing her research work in Switzerland, where she was working on different types of benzoic acids and their isomers, which was, of course, at the time, one of the extremely hot topics. Now I just want to look back to Gustav Kompa for a second here, and I want to show kind of the coolest total synthesis that Finland has ever produced. At least this is how we perceive it. So Kompa was the first. And this is kind of what made him world famous, and this is also what kind of initiated organic synthesis research and organic research in Finland altogether. Gompa was the first chemist to basically crack how camphor should be made in the lab. This is the original chemistry, the publication in 1903. This came out. So the route Gustav Gompa, and mind you, for everyone who's used to modern chemistry, he didn't have access to NMRs. He didn't know what flash chromatography is. They couldn't TLC stuff. And this is what they did. So you take this oxalate, you condense it, you get the cyclopentanone intermediates, right? You methylate it here. There was some back and forth between if it's actually methylated on the carbon or oxygen, proves that Gompa was right. This is the compound. Then you just reduce it, treat this with HI to get an olefin. Nowadays, of course, we would just hydrogenate this off. Back in the day, you couldn't hydrogenate it. So Gompa basically just brominates the double bond, uses zinc to cleave off the carbon-bromine bond. With acyl chloride, you can actually turn this into an internal anhydride, and then you can hydrolyze that anhydride into camphoric acid, and this basically completes the synthesis. The final steps to reach camphor from this camphoric acid are known, so you basically just need a homologation step and so on. And this is a monumental piece of work, especially considering that this was carried out in Finland, where we kind of had to ship everything from Germany. And this was also congratulated by William Henry Perkin Jr. himself by stating that we really don't want to continue our total synthesis of camphor because it's much less satisfactory than what Gompa has published. And these were kind of the beginnings of the process. And eventually this Gompa synthesis, this total synthesis, he built a factory in Finland, and they produced some 200 kilos of camphor using this type of synthetic chemistry in Finland. And these were kind of the starting points of modern chemical industry in Finland. Well, things have moved forward. So what's currently happening in Finland in terms of organic synthesis? Well, there's a lot of fine chemical production. We are heavily involved in nanochemistry in all sorts of facets, and also we're heavily involved in biochemistry, and you will see this with today's presentations as well. Just to give an idea, here are kind of the universities within Finland. The locations are kind of off. Don't worry about this. These are kind of just roughly placed wherever. The university that carry out research in organic chemistry. So up north, we have the University of Oulu, where they're especially looking at renewable chemistry. We have Tampere University, which is extremely big on applications in materials chemistry. We have University of Turku and Åbo Academy. Both are working on extremely multifaceted areas of organic chemistry. And then we have in the capital region, Aalto University, where I'm located, and University of Helsinki. Both as well have kind of multifaceted areas of organic chemistry being studied. In central Finland, we have University of Jyväskylä, where I got my PhD. We also have people from there. They're very, very big on nanochemistry and supramolecular chemistry. And University of Eastern Finland, where pharmaceutical chemistry is one of the key things that they are actually studying. The key fields, or the major research themes, like within the entirety of Finland, kind of split themselves into functional molecules and materials, all sorts of surfaces, further modifiable things, supramolecular machines, sensor units, these kinds of things. So molecules with a function. We do quite a lot with chemical biology. This includes total synthesis of bioactive natural products, bioactive compounds, the study of their biology, signing bioprobes, and so on. And then, of course, well, being heavily dependent on forests, we're also looking at biomass valorization and kind of developing circular economies. So if you just get lignocellulosic material, how can you actually apply that lignocellulosic material in kind of more sophisticated ways than just making paper? Can you actually turn that into fine chemicals and so on, which is definitely a big challenge for organic chemistry. It's of course not only universities doing this research. We have plenty of companies. This is just the few of the biggest ones. We have plenty others. Within Finland, kind of the key statistics are that, excuse me, around 20% of all of our exports come from chemical industry. So we are one of the biggest industries in Finland. The investments hang around 1.3 billion a year, R&D investments around 500 million a year, and the exports rake in around 12 billion a year. Of course, we have Metsä Board, EuChemS, Stora Enso, these kinds of big forest companies or forestry companies. We have Neste, who are now looking at biofuel production, Orion as one of the biggest pharmaceutical companies who are developing their own drug molecules as well. We have companies working on surfactants, paints. We have Fermion and CAB, for example, both working on industrial scale organic synthesis. With that said, today's presentations are going to be coming from University of Turku. We have Anu Airaksinen, then I'm going to shut up soon. I'm from Aalto. From University of Helsinki, we have Philip Ekholm and Jan Deska. Then our company representative is Toni Metsänen from Orion. Then University of Jyväskylä, we have Kaisa and Petri. And then University of Eastern Finland, we have Petri Turhanen. With that said, that's all I had. Yuri, should we move on with today's presentations?
Thank you, Juha. It was very brilliant. I think if there's no any questions, we can move on. I think the next speaker already ready. I guess we don't have questions, so maybe we just continue. So our next speaker is Professor Anu Airaksinen, who got her PhD from the University of Kuopio in 2003. Nowadays, this university is known as University of Eastern Finland because it's was united about 10 years ago with University of Joensuu. And Anu is currently working in the University of Turku Pet Center, and previously she was working in the University of Helsinki around 10 years. So Anu, please continue from here.
Thank you very much, Yuri, for this kind introduction and invitation to come to talk to this great event. I will start sharing with my screen. Just a minute. Sorry for this. So now. Just a minute. So there is this one on front of that. Okay, so up we go. So now we have the presentation on. So it's great pleasure to be here talking to you and introducing topics in synthesis of pet radiopharmaceuticals and our race against time. Radiopharmaceutical chemistry and synthesis of radiopharmaceuticals, it's not only happening in our unit at Turku Pet Center. There are also other locations in Finland where pet radiopharmaceuticals are synthesized. To mention, in Kuopio University Hospital, there is clinical production of pet radiopharmaceuticals as well as in Helsinki University Hospital. There is also really active research group at Helsinki University led by Professor Mirkka Saarparanta called Tracers in Molecular Imaging. Turku Pet Center, so it's a national research institute for pet imaging and for use of short-lived positron emitting isotopes in the field of medical research. It's owned by 3 parties, University of Turku, Turku University Hospital and Åbo Academy. So currently we are producing 40 GMP level radiopharmaceuticals for clinical imaging studies. And in addition to that, we have over 40 preclinical tracers which are produced for preclinical experiments in different animal models. And this naturally this list of the preclinical tracers, this is something which is living all the time because new tracers are coming for testing constantly. So the pet radiopharmaceuticals which we produce and the tracers which we produce, so we are sourcing the clinical and preclinical imaging facilities. We have several scanners, 8 PET scanners in total, from which the newest one is the total body PET CT Quadra scanner, which was just recently installed last spring. So the imaging facilities which we have, so we are contributing to Eurobioimaging European infrastructure and providing the research services for researchers all over the Europe. The main research focus areas at the Turku PET Center are cardiovascular and metabolic diseases, neuroscience, cancer, preclinical and translational research, and radiochemistry research, instrumentation, and data science. So for the radiochemistry research, our key focus areas are in development and production of radionuclides and radiotracers with high quality, quantity, and high molar activity. And what is important for radiopharmaceutical synthesis and production is the development also the automation methods for radiosynthesis and quality control procedures, and also to develop the radiometabolite analysis of new tracer candidates, which is needed for especially for clinical translation of the new tracer candidates. For PET radiopharmaceutical research for production and also for synthesis of investigational tracers, we have facilities with 3 cyclotrons and 3 gallium generators, which are used for production of radionuclides for the studies. In total, 26 hot cells in 3 different locations, and then also organic synthesis laboratories for synthesis of precursors for radiolabeling. And the radiosynthesis laboratories, they are equipped with automated synthesis devices, and they are next to quality control laboratories for quality control of PET radiopharmaceuticals and pharmaceutical candidates. So as you saw, so we at the moment we have a really large selection of PET tracers for human and animal use, almost up to 100 tracers in total. So the radiochemistry research which we have at the PET Center, so we belong to Department of Chemistry at University of Turku as well. And we are not the only group or team working with organic chemistry or synthetic organic chemistry at the Department of Chemistry. So here is some of the groups to highlight some of the research which is carried out at our department. To mention Pasi Virta, Professor Pasi Virta, who is head of the department. So he's working with DNA-based drug delivery systems. Professor Tuomas Lönnberg he's working with metal-mediated base pairing. Dr. Harri Härmä with label-free protein recognition, and Professor Karita Kvarström with conducting polymers for functional polymers and composite films. So one of the strategy focus areas of the University of Turku is health diagnostics and drug development, and we are greatly contributing on that strategic focus. It's good to notice that we have international master degree program in physical and chemical sciences. So we are really happy to accept international students to study chemistry at our department, and also we have a doctoral program in exact sciences for our doctoral students. So what are radiopharmaceuticals? So simply they are compounds which are labeled with radioactive isotopes. They can be used either in radiotherapy as radiotherapeutic agents, or they can be used for nuclear imaging as radiotracers, as radiopharmaceuticals. For positron emission tomography, radiopharmaceuticals, they are labeled with positron emitting radioisotopes. And the whole imaging is based on detection of 2 isoenergenic gamma rays, which have energy of 511 kiloelectron volts. And this makes the positron emission tomography highly sensitive and truly quantitative imaging modality. So if you look on the radionuclides which are listed here in this table, these are typical radionuclides for positron emission tomography. Oxygen 15, nitrogen 13, carbon 11, fluorine 18, and gallium 68. So you can see that the half-lives, the physical half-lives for these radionuclides, it's really short. So we are talking about radionuclides with half-lives of minutes to up to a few hours. And this is something which really gives special features to our work in synthetic organic chemistry. So how the PET actually works. So when we have the proton-rich nuclide, which is it's not stable, so it decays by emitting a positron. And this positron, when it travels in tissue for a certain length, it's called positron range, which is dependent on the energy of the emitted positron. So when this positron encounters an electron, so there is an annihilation of the energy of these two particles emitting two gamma rays with the same energies, 511 kiloelectron volts, to the opposite directions, and this can be then detected with the PET scanner, and from there we can reconstruct the image. PET imaging is highly sensitive method, like I already said. So it's really we can detect even femtomoles of the injected tracer. And with PET, we can investigate fate of the tracer without interfering the biological system itself. And it means that due to the sensitivity, we can even investigate compounds which are highly potent or they are really toxic, and we can study them in amounts which are subpharmacological or subtoxicological. But this is only valid if we have high molar activity for the radiopharmaceutical. What is this molar radioactivity? So this is something which is really important when working with radiopharmaceuticals and when working with radiosynthesis, because this is something which you need to take care of from the beginning to the end. So the unit of the molar radioactivity is gigabecquerels per micromole. And if you look on some of the positron emitters listed here, so the theoretical molar activity it's really high, top high. So we are talking about from 1,000 to several hundred thousand gigabecquerels per micromole. In practice, these cannot be achieved because there is always some dilution due to the stable isotopes, and also the time matters, because the time influences naturally the molar radioactivity as well, because the molar radioactivity as a activity per molar unit, it decays following the decay law exponentially together with the radioactivity. Also, this is the reason why the time matters. How the molar activity actually affects the PET imaging. So it affects it a lot, because when doing PET imaging, we need to fulfill the tracer principle. And there is this is a really good example of that. This was published already in 2004 by Hans Olsson from Karolinska Institutet. So he studied PET imaging of human brain by using carbon-11 labeled FLP457. And here you can see a human brain, the coronal plane of human brain, and on the left side you can see the PET image when high molar activity radio tracer was used, and on the right side you can see when low radioactivity tracer was used. There is a clear difference, because with the high molar activity tracer, we can see much more intense radioactivity in brain, radioactivity accumulation in the brain, than with low molar activity tracer. And this is due to that reason that simply when using a low molar radioactivity tracer, there is some stable isotope labeled tracer which is saturating the binding sites, where also the radioactive derivative is going to be found. So the tracer principle is not valid if the molar activity is too low. But how the high molar activity influences the radiosynthesis of radiopharmaceuticals. So it affects the reaction kinetics, because usually the radioactive starting materials which we are using, so they are on nanomolar nanomolar amounts. When using the other reagents, when using the catalyst, they can be in micromolar range. So there is a clear difference in stoichiometry in this case. Also, the solutions reaction mixtures, they are really diluted because usually we are anyhow working with hundreds of microliters to a few milliliters of reaction volumes. So the radioactive starting materials are really diluted in the reaction. Also, an adsorption is an issue because when working with such a tiny little amounts, there is lot of loss which may occur simply due to adsorption to different type of materials. Also, characterization can be sometimes challenging because due to the low amount of the final product which we receive, so it is not enough for any NMR characterization. So we need to rely on mass spectrometry and HPLC characterization methods. One big difference to organized synthesis with stable isotopes is the limited selection of radioactive starting materials which we have at hand. Many of the PET radionuclides, they are produced in-house by using cyclotrons, so little particle accelerators. And it really depends on the cyclotron production in which form we will get our primary precursors where we can start from. For example, for fluorine 18, we can get the fluoride 18 as a anionic fluoride in water solution, or then if there is a production of electrophilic fluorine, so then it's in fluorine 18 F2 gas, which is significantly diluted with the stable isotope F2 gas. For carbon 11, for example, the carbon 11 can be produced with carbon 11 carbon dioxide or carbon 11 methane in target. The short half-life itself also causes its challenges because due to the short half-life, we need to take care of the time which we need for the synthesis and for purification and formulation of the radiopharmaceutical. So that means that usually we need to start with really high starting radioactivities, varying between 1 gigabecquerel to several hundred gigabecquerels, depending on the study where the radiopharmaceutical is needed. And this is, of course, really demanding also during the radiation safety aspects of the employees, so that we need to take care of the radiation safety of our staff. So usually remote controlled systems or fully automated radiosynthesis units are used in lead shielded fume hoods. The short half-life means also that the reaction times which we are reactions which we use, they need to have short reaction times. We need to be able to try the reaction to completion within a short time, and also limit the number of sequences, so the late stage labeling procedures are preferred. Also, we need to consider automatization of the whole synthesis sequence. So for that reason, 1 pot, 2 pots, and different kind of inline procedures are preferred. One big difference is reproducibility, because our product disappears. So even though the synthesis worked today, so it must work also tomorrow. So we cannot store the product. So it just disappears. So therefore, it means that the reaction which we have optimized, so it needs to be reproducible. We need to be sure then that we can reproduce the same conditions for the reaction day after day, every time. So then to some of the highlights for the research at the PET Center in Turku. So Dr. Anna Kirjamainen, she has been working with utilization of transition metals in fluorine 18 radio labeling chemistry, and this is for the late stage fluorination of radiopharmaceuticals. This is a really important topic in that sense that especially electron-rich arenes, they have been really difficult to fluorinate by using fluorine 18 fluoride. And if we consider the production of fluorine 18 fluorine, there is 2 different methods for cyclotron production. One is this production of fluorine 18 fluoride in aqueous solution. Another one is fluorine 18 electrophilic one in F2 gas. For the electrophilic production, production, it requires addition of significant amount of carrier fluorine-19 gas. So that means that when producing electrophilic fluorine, we dilute the primary radioactive starting material by a stable isotope, and that way decreasing the molar activity of the starting material. And this has been a significant challenge in our field, so that how to efficiently fluorinate aromatic positions, especially when they are electron rich with nucleophilic fluoride. And this fluorine-18 deoxyfluorination of phenols, which is presented here, so that was originally published by Tobias Ritter from Harvard University in 2017, and here we can see that it's based on the formation of a ruthenium phenol complex, which is then fluorine-18 fluorinated in presence of chloroimidazole salt. And here really like elegantly Anna has utilized this reaction for production of fluorine-18 FP-ATPP, which is a CB1 cannabinoid 1 binding radiopharmaceutical and has been really difficult to produce otherwise. By using this method, Anna was able to produce the fluorinated radiopharmaceutical with excellent or good radiochemical yields, 16.7%, and also with high molar activities with 95 gigabecquerels per micromole, which would have not been possible when using electrophilic fluorination methods. Anna has been also working with fluorination of arenes with several different approaches, and here really nicely she presents the fluorination of NS12137, which is a norepinephrine transporter binding radiopharmaceutical. So that and there is also this electron-rich position in the aromatic ring, which need to be fluorinated. And this compound has been traditionally radiolabeled with F2 gas with low molar activities, but by other methods, starting from fluorine-18 fluoride, nucleophilic fluoride, it's possible to radiolabel it with higher molar activities. And here there is 3 different approaches which have been compared. So there is fluorine-18 F2, which was produced by a method, post-target synthesis method, originally developed by Professor Emeritus Olof Sulin, which is where the F2 gas is produced in this type of electron chamber from the fluorine-18 methyl fluoride, and this is then used for direct fluorination with 3-methyl stannyl precursor presented here. Another approach is to use the F2 gas, which was produced from the nucleophilic fluoride for synthesis of select fluor electrophilic reagent, and synthesize the final product or the intermediate, in this case, from the same precursor by using the electrofluorination reagent, or then for using copper mediated fluorination directly to this stannyl precursor. So this direct method was originally reported by Peter Scott from University of Michigan, and you can see here how excellent is it compared to other methods in terms of molar activities. So with this copper-mediated reaction, 300 gigabecquerels per micromole molar activities were achieved. It's not always possible to use late-stage fluorination for compounds which are really sensitive, especially when we are talking about biological compounds like peptides, antibodies, antibody fragments, proteins, and so on. So then other approaches need to be applied. So here is results of my doctoral student Sofia Otaru, who is finalizing actually her doctoral studies at University of Helsinki soon and have her defense in 2 weeks. So she developed the fluorine-18 labeled tetrazine, which has a Twitter ion here in this position, and used that for radio labeling a TOC peptide octreotide by using inverse electron demand disalder ligation. This ligation it was originally developed by Caroline Bertozzi and introduced to the nuclear medicine field by Raphael Rossin and Marc Robillard. And here we were able to demonstrate how this small peptide was efficiently radio labeled by using this fluorine-18 labeled prosthetic group in short reaction times and in physiological conditions. And because this is bioorthogonal chemistry, so really simple purification and formulation could be applied. The similar strategy has been utilized by my student Tatjana Ovchinnikova from Turku PET Center, where she have radio-labeled spherical nucleic acids, which have been developed for theranostic agents in collaboration with Professor Pasi Virta from the Department of Chemistry from University of Turku. And so these spherical nucleic acids, they contain this fullerene core, and which is surrounded then with the oligonucleotide sphere, which has then conjugated the tissue-specific ligands for targeting of these SNA compounds. And Tanja used fluorine-18 FDG tetrazine for conjugation for radio labeling of these SNAs with different kind of structures really efficiently by over 85% radiochemical yields and radiochemical purities over 99%. So with this method, we were able to compare how the different structural modifications on these spherical oligonucleotides affect their biological properties and their biodistribution in animals. And here two different backbone structures were evaluated, either based on phosphodiester bonds or phosphotioate bonds, and then also a targeting moiety folate for tumor targeting. And here we can see results from the biological evaluation. So how here I ask you to focus on the black and green marks, so that there we can compare directly how the phosphotioate backbone affects the biodistribution. So the blood circulation time is significantly prolonged compared to the phosphodiester backbone SNAs, which can be also seen here nicely from the animal images. Assistant Professor Xiangguo Li, so he has been working with click reactions as well, and here is a good example on radiosynthesis of fluorine-18 fluoromannan for PET imaging of mannose receptor in inflammation. In his work, Xiangguo, he synthesized the fluorine-18 labeled bicyclonone, which he really used efficiently for radio labeling of mannan. So you can see here the reaction occurs in room temperature in 3 minutes. And really nicely, here we can see how the atherosclerotic plaques in aorta have accumulated the tracer on the aorta wall here. As a collaboration with Xiangguo, so we have developed also fluorine-18 labeled FDR, so which was efficiently synthesized from the corresponding tosylate precursor with 60% radiochemical yields. And this FDR can be used for oxime ether formation with amino-oxy functionalized peptides when catalyzed with aniline. And with this method, we have been able to produce peptides with radiochemical yields varying from 27 to 37% with good molar activities. Here is an example of fluorine-18 labeled FDR cyclic, which is accumulated nicely on the inflammation site when injected to an inflammation model. Okay, so as a summary, I would say that so the short half-life and high molar activity. So these are these typical features of positron emitters, and these features really affect the synthesis of PET radiopharmaceuticals, and usually they cause some challenges. And certainly we need the development of new synthetic methods and procedures for more efficient production of PET radiopharmaceuticals. And in this sense, we need to remember that time is one of the variants. So time is one of the factors which is influencing the radiochemical yield and the molar activity of the final products. And the method which we select for synthesis of radiopharmaceuticals, it affects the in vivo performance of the tracers. So if the molar activity, for example, is low, so certainly this is something which we can immediately see in our PET images. So there are many people to acknowledge, of course, from all these results which I just presented to you. So I would like to thank Professor Mirkka Sarparanta's team at University of Helsinki, and then at University of Turku, Professor Sirpa Jalkanen and Professor Pasi Virta and his team, and the staff of Turku PET Center certainly, and especially our lead of clinical radiopharmaceutical production, Sarita Forsbacka. Thanks to her, so we have such a great repertoire of clinical radiopharmaceuticals in-house. And if you are interested to join us, so just send me an email, so you can find my email address here from this slide. We are happy to recruit new doctoral students and postdocs at any time. And thank you for your attention. I'm happy to answer any questions if you have any.
Thank you, Anu. That's the whole presentation, off to a great start. Thank you very much. There's one question already in on the Q&A, which is also you can use to ask a question from Dr. Mahadio Patil. How about the scope of radioisotopically labeled deuterium or tritium for PET study? And what are the requirements of specific activity of the radionuclides for PET study?
Okay, so deuterium and tritium, so they are not positron emitters, so they cannot be used for imaging in that sense. But the deuterium is frequently used actually for modulating the metabolic stability of radiopharmaceuticals, because it may affect the how quickly certain positions are enzymatically metabolized, and so in that way deuterium is utilized also in PET radiopharmaceutical chemistry. And Great. The requirements of specific activity, now I can see there is an additional question. Yeah, so I would say that as you could see from the list, so there is iodine 124. It has the molar activity approximately the theoretical 1,000 gigabecquerels per micromole, and this is adequate, but certainly higher the better, because in that way we can maintain the tracer conditions.
Great, Annie. Oh, there's another question coming through from Ilan Marek. Oh, am I the only one who has a bad connection? I don't know, Ilan. I can't answer that. Hopefully everyone else's connection is okay. So I can't deal with that one. Again, questions from the audience. And again, with Zoom, just use the raise hand function, and then I will unmute you if you wish to ask the question yourself. Alternatively, as we've had from the first example, we can have the question and answer typed in, and then we can read it out. Maybe Anu, I could ask a question. So what are the key challenges still remaining for the development of radiopharmaceuticals? So what still has to be solved?
which still has to be solved. So there is so many things to be solved. So first of all, it's I would say that the translation of new tracer candidates to clinics, because it is possible to use quite complicated and multi-step synthesis for preclinical experiments, but translation to human studies is not always that simple. So for developing methods for smoothing the translation.
Yeah, late stage functionalization is really where it's all at.
Yes, late stage functionalization, yes, indeed.
Yeah. Okay, so perhaps in the interest of time, that's a half hour. Thank you again, Anu, for that superb presentation. And I'll pass over to Yuri, who's going to introduce the next speaker.
Thank you.
Okay, thank you. It was very nice presentation. And our next speaker is Petri Turhanen, who also got his PhD from the University of Kuopio in 2003 and has continued his research since then at Kuopio. Currently, Petri is working as a senior scientist at the School of Pharmacy at the University of Finland, Eastern Finland. So please, Petri, tell us about your research.
Okay, maybe I need to share my presentation. Can you see and can you hear me?
Yes, perfect, Petri, super.
Okay, then, yeah, let's continue. So this is what I have done around 25 years. We stepped to the organophosphorus compounds and organophosphorus chemistry a little bit, I think so. So yeah, the title synthesis of bisphosphonates and ATP analogs, meaning adenosine triphosphate analogs. And then there's gonna be some brief introduction to that Dryad-Dowex method which we have found. So let's go. Okay, something about bisphosphonates. Okay, they are the analogs of naturally occurring pyrophosphate, which can be found in all mammalian cells. They are just the stable analogs of those. And the first bisphosphonate was prepared on 1860s, something like that. I don't remember what this was, but maybe the medronate or editronate, something like. Bisphosphonate as bone targeting agents were discovered late 1960s, and then this has been continued, the research, quite intensively. There's you can see the structures. There's a pyrophosphate, there's a phosphate structure, phosphate where you have in the middle oxygen, and everybody can imagine that this is not stable in the enzymatic form, enzymatically. But when we change it to the carbon, then it's come to be stable, enzymatically stable, and there's a lot of variability here in R1 and R2 groups. There are some traditional uses for the bisphosphonates. At the beginning, they were used in industrial purposes, and they are used still in industrial purposes. Corrosion inhibitors, and water softening. And typical examples of the medicinal use are those osteoporosis drug. Here you can see Fosamax, which is the alendronate bisphosphonate, which is used to the osteoporosis. Then there's also format that anti-tumor agents, they work, drug delivery and targeting agents, and also radiopharmaceuticals. They can form chelates with the metals, and they have used for the bone imaging with the technetium-99. I think Anu will know that. anti-inflammatory agents also, and so on and so on. There are lots of medicinal use for the bisphosphonates. But yeah, there's then some examples of the bisphosphonates which are in clinical use. There are non-nitrogen containing bisphosphonates. This is medronate, which is the most simple one. Then there's the etidronate, which is the most maybe the most simple one, hydroxy bisphosphonate, which has the hydroxyl group in here in the middle carbon. And then there's the chlordronate, where is two chlorines in the middle carbon. These are the first generation bisphosphonates. Then there are nitrogen containing bisphosphonates. There are a couple of examples. There's the alendronate, which has 3 carbon chain, and then there's an amino group here, and then it's a hydroxy bisphosphonate. Then there's a pamindronate, which have 2 carbon chain, and then the amino and hydroxy, also hydroxy bisphosphonate. These are second generation bisphosphonates. And now there are also the third generation, and then meaning that they have some nitrogen in the aromatic ring or whatever. This is cholendronate, which has this kind of backbone structure. Then there's the risendronate, which have that kind of backbone structure. And then there's a difference between first generation, second generation, third generation. Actually, nowadays there are the fourth generation also, but I have not picked up them here, the examples. There's a difference that the second generation bisphosphonates are around 10 to 100 more potent, like in osteoporosis, and third generation are then some 100 and 1,000 more potent than the second generation bisphosphonates. Actually, most potent one I know is this cholendronate. You need a very, very small amount to treat the osteoporosis when you use the cholendronate. So, some properties of bisphosphonates, why they are so good in many ways. There's the PCB structure which is enzymatically and chemically stable units. In some cases, depending on the R1 and R2 groups, which coming starting from here, it may be not so stable in chemically, but enzymatically it's totally stable, and that's why they have found this is used for the like osteoporosis drug. Okay, bisphosphonates are extremely good metal chelators. They form that 6-membered ring, which is a very favorite structure energetically, yeah. And clinically, these bisphosphonates are used. Clinically used bisphosphonates are especially good chelators of calcium, and bones are mainly hydroxyapatite. So there are a lot of calcium in the hydroxyapatite, and meaning that you have a lot of calcium in the bone. Then the properties of bisphosphonate can vary a lot, and mainly they are depending on the bridging carbons to substituents. This hydroxyl group increase a lot of metal affinity. And another group here, it's rule out, of course, the solubility. Is it water soluble or is it not? Or biological activity, and also the metal selectivity. Which kind of metals are selectively bind to the bisphosphonate structure? Well, then some synthesis. So the basically the synthesis of 1-hydroxy 1,1-bisphosphonate. Basically, you can do almost all kind of those using this Kieczykovsky method, which has published in 1995. And you just use the starting material, carboxylic acid, then treated PCl3 phosphorus acid in methyl sulfonic acid and C65 70 Celsius overnight, and then adding water reflux 4 to 16 hours, and basically then you get hydroxybisphosphonate. And only thing to purify is that you precipitate them. You can precipitate them either as acid form, or you can precipitate either as salt form. That depends on the structure of the starting carboxylic acid. Like using sodium hydroxide or potassium hydroxide, whatever. Then there's a method, if you cannot make a bisphosphonate, hydroxybisphosphonate using that Kierzykowski method, then there's this method which was published in 2011. So you can use the carboxylic acid as starting material, then catechol-borane forming hydrogen, then activating this carboxylic acid, and then using tris-trimethylsilyl phosphate, and liberating then the decellular the material with methanol, you get the hydroxybisphosphonate. This is the one way to do it. And then there's this also that you activate the carboxylic acid at making that carboxylic acid chloride, and then use the same method to get the 1-hydroxybisphosphonate. Then there's this most oldest one method that you use the carboxylic acid chloride, use the 3-methyl or 3-alkyl phosphate, made a phosphonate, acyl phosphonate, and then adding the dimethyl phosphate, catalyzing by base, you get the tetra ester of 1-hydroxybisphosphonate, which you can liberate, of course, with concentrated hydrochloric acid, refluxing it some hours. You get the then but this thing make you a variability to make also the partial esters of 1-hydroxybisphosphonate, like D-ester, 3-ester, or this kind of D-ester. And this as here. Some other information which related to the bisphosphonate. As I mentioned, the bisphonols have wide range of medicinal application, but they and in that case, in that point of view, they are not just osteoporosis drug. There was those which I already mentioned, where the bisphosphonate can be used. But then there are a lot of applications which are non-medicinal. It can be used in agriculture as fertilizer, in food science as antioxidant, in metallurgy as surfactants for metal, scale inhibitors, corrosion inhibitors, in chemistry as chelating agents, catalysts maybe also, and of course ligands for metal organic frameworks. In polymer chemistry as modifiers and additives, in environmental chemistry as wastewater purification agent. As here you can see this N10O, which we have synthesized in our lab already in 1 kilogram scale, and it is very effective for the metal ions removal from aqueous solutions. It works very nicely for the example scandium. When you have a scandium diluted acid solution which pH is around 0, you can still get that scandium out of that solution with using this N10O. It's very effective. Yeah, and in our lab, I have synthesized that more than 160 different kind of bisphosphonates or its derivatives, or under my supervision. they have been synthesized. There are actually a lot of Erasmus students and which have been done synthesis work with bisphosphonates. Okay, then we jump to this adenosine triphosphate analogs, and they are important because these nitrogen-containing bisphosphonates induce the formation of this kind of structure, adenosine triphosphate analogs. Yeah, basically the use of those non-nitrogen bisphosphonate and nitrogen bisphosphonates leads to the inactivation of osteoclasts, which degrade bone, but they have the different mechanisms. Here it's a picture for it. And then when the use of the nitrogen bisphosphonate like alendronate here, it's induce the formation of APPI and also the APPPD, which have several biological functions. They are really important in biological. They are formed in your cells. Well, there was a lot of need for that APPPI. And there was previously only one method reported in literature, and there was only 1 to 2% yield. And of course, it was purified some HPLC method. So there was a need for better methods. And we found the better method, which has published in 2015. And then there was 2 strategies how to synthesize this APPPI. Yeah, this method, which we published, led to the tetraalkyl ammonium salt of APPPI, which is extremely hygroscopic. And then there was again a need for the more practical form of APPPI. So we developed another method, leading a little bit better yield. These are actually good yields for this kind of structures. As you know, they are pretty unstable, these phosphorus oxygen phosphorus bonds. They need to be, because they need to be degraded in your body by enzymes. So we found the method, and also we used quite not so common purification method. We used the high performance Gaussian current chromatography, and get it a very nice yield and pretty nice yeah, okay. Yeah, we started from the ATP, and then we used the DOX H+, which is cation exchange resin, and then formed to the tetrabutyl ammonium salt of the ATP, and then tosylated isopentenyl alcohol was introduced, and then stirred some time, and then purification with HPCCC. And yeah, we get 20% yield, and it can be 50 milligram in one batch after conversion to the sodium salt form. And this is not a hygroscopic one. And then synthesis of APPD. It was requested from Professor Eric Oldfield on the University of Illinois. He is the top scientist in the field, which I know, in this organophosphorus ATP analogs and bisphosphonate also. And he was asking, because he was asking for me that can I send him some APPPI, and can I synthesize the APPPD? Okay, I just told him that I can try. It wasn't the easy one, but I finally found ATP disodium salt. Again, change the cation exchange, change the sodiums to the hydrogen, and then tetrabutyl ammonium, get the ATP tetrabutyl ammonium salt, and then adding the silver carbonate in acetonitrile, we get silver salt of the ATP, and then adding this 3,3 allyl bromide, and we get the APPPD. So and also, of course, because it's the same kind of structure than APPPI, so it was purified by HPCCC and get it 21% yield. Yeah, and it was published in ACS Omega 2017. And why? Yeah, the APPI and APPD, because I have them now in my storage, and that's better availability. Those ATP analogs led to the several collaboration projects, and results are publications, of course. Selective calcium-dependent inhibition of ATP-gated P2X3 receptors by bisphosphonate-induced endogenous ATP analog. Yeah, it was published in 2017, and meaning that this API has a straight effect to the pain relief. So, and then there was another publication, 2018 collaboration publication, and then this bisphosphonate generated ATP analogs with Eric Oldfield in EuChemS 2018. Sorry. Yeah, okay, then I have done the synthesis of de-deuterated APPPI, because it's very potential mass spectrometry standard for quantitative measurement of API from biological samples. I think there's a real need for that. At least earlier there was a need for that. But let's see, I have submitted the manuscript, and it was, yeah, there's the 2 deuteriums here, so it should be potential mass spec standard. Okay, then we jump to this DOWEX method. This was found by chance when we were preparing the phosphate ester precursor for this ATP analog synthesis. Okay, so yeah, we started from actually, I synthesized this phosphate, 3-phosphate precursor, and then this is a normal reaction to sodium iodide used in acetone to remove selectively one methyl group in the ester. Then there's used 2 DOWEX again to change sodium to hydrogen, and then we, with Professor Webseläinen, we just found out that there was a format also this kind of compound. So this addition of the iodide in that tertiary position was observed, which is very uncommon reaction. So the commercial DOWEX contains a lot of water. When you buy it, it's something like 60%, even 55 to 60% it's contain water. So there was a need for trying it. Actually, nowadays the DOWEX is not a DOWEX. Trade name is not DOWEX anymore. It's the Amber Chrome. When I last time ordered it, there came Amber Chrome, something like. They have changed the name. But yeah, it can be dried in vacuum, but it takes several days to get it totally dry, and there was a bubbling and a lot of problems. Then we just try to dry in oven, and found that it's the reasonable way, very effective, and several tens of grams can be dried at the same batch. So there was you can see there's a DOWEX in its water containing form, and then there's a DOWEX with after the overnight drying in the oven, 120 Celsius. Yeah, this was quite a surprise, because the DOWEX is basically the resin, which is formed, well, it's the plastic. Yeah, I don't just now remember the. Okay, but yeah, this method. Here's the scope of the method. Some examples where in all these reactions have used this dried Dowex. We can esterify like oleic acid in 15 to 1 hour using Dowex and a catalytic amount of sodium iodide in room temperature. And then we can trans-esterificate. We had a sample of real used cooking oil, and then we refluxed overnight and get the first generation biodiesel. So then there was a carbon. We used the starting material, and in room temperature 2-hour reaction, one equivalent of sodium iodide in acetone, 45% addition to the tertiary position iodide. Then if the same reaction was left on overnight, then the aromatization was observed. Then there's a double iodine addition to the propalkyl alcohol, 65 Celsius overnight, in 2-propanol, getting nice double trans product. And then there can be also substitute the hydroxyl group selectively with a reflux 5 hours, 2.1 equivalent sodium iodide in acetonitrile. Nice product. Then you can also use the starting material, the sugar, the glucose. So it was it was quite nicely esterificate to the again substitution reaction, and then we add that to propyl group to isopropyl group here in hydroxyl, this primary hydroxyl group. Okay, this has this Dowex research has led a couple of publications. This was the first one, actually, some kind of a proof of concept which we published in 2015, and then 2016 another one. Preparation using building blocks from the cyclic ethers. This was quite a nice. And then there's that green and efficient esterification method, which we published in 2019, and then this has got a lot of views in the American Chemical Society pages. It's over 11,100 right now, and meaning that there's still the esterification reactions have a kind of attention. I don't know why. Okay, and then with Yuri we have published this deiodination method to prepare trans deiodinated products starting from the alkenes. Okay, I think I'm gonna be almost ready. So the collaborations, of course, Professor Jouko Vepselän is my earlier supervisor and mentor also. And Professor Epi Lehto, we have the collaboration with bisphosphonate modified mesoporous silicon. There are several publications with that. And then the maybe most important collaborator right now is the Professor Kostas Dimadis and his group in University of Crete. I made bisphosphonates, and he's doing the research with controlled release and metal organic frameworks with bisphosphonates. And then from his groups is another Kostas Papathanassiou. He's now in University of Wolverhampton, and also the controlled release studies. And Jukka Leppänen and Juri Timonen, of course, this dry drawers method, and of course Pavel Kafarski need to be mentioned because his inspiration have been lots of me, because he's the one of the leading guy in the organic phosphorus chemistry and actually the amino bisphosphonates and bisphosphonates generally. And then goes to Marita Salminkoski, retired. He's helped me a lot in technical. And then, yeah, if you have something, if you need a bisphosphonates, if you need something, just send me an email. Okay, maybe I'm done.
Super, Petri. Great talk. Learned lots about bisphosphonates and new and exciting applications for DAWIX. So there's a question from the chat from Shengying Saya. Thanks for the talk. Amazed by the color change of the forms of DAWIX. And the question is, will the dried DAWIX black turn back to the light orange color form when wet?
No, it don't get back. It is still that quite brown and what I don't know why. I just was thinking about what it is that the DAWIX, because I just can't get my head right now. It's the resin which is Yuri, tell me. Yeah, it don't get back in orange.
Okay, we have a chat, a call coming in from the chat from Lawrence de Cone. So, Lawrence, you should be able to talk now. Lawrence?
Yes, do you hear me?
Yes.
Yeah, so I'm wondering if the DAWIX plays a role by shifting the equilibrium by taking up water. Do you think that happens?
Yeah, that's one possibility, of course. Yeah, it can take the water. We have a think about that, and we have a lot of studies, or not the studies, but we are proposing a lot of reaction mechanisms why DAWIX is doing that kind of things what it is doing. There are, I think there are, this is a one point of view. It can take back water, I think, in somewhere in its structure, and then there are maybe also this well, yeah, okay, there might be also other reaction mechanisms which are involved. Not coming in my mind now, because yeah, we have thinking about that, and there's a couple of at least 2 publications coming, and then we have a manuscript in preparations where we take account to the why the DAWIX is working like it's working. There might be, yeah, okay, sorry.
Yeah, I'm looking forward to those publications. It's a really interesting work. Thank you.
Okay, thank you.
Thanks, Lawrence. Again, any further questions? Just looking at the time, maybe in terms of the schedule, we might keep going. But hard rock, hallelujah, Petri. Well done.
Okay, thanks.
Okay, over back to you, Yuri.
Thank you. And thank you for the presentation, Petri. It was amazing, because I haven't seen all those 3 different things you are doing all together. So our next speaker is Dr. Philip Ekholm from the University of Helsinki. He got his PhD in 2012 from the Åbo Academy in Turku, and worked a couple of years in industry before moving to the University of Helsinki. And currently he is working as a group leader and also holds adjunct professorship both in the University of Helsinki and Åbo Academy. So please continue from here, Philip.
All right. Thank you very much, Yuri, for the kind introduction and also for the invitation to present some of our work here on this forum today. Can you guys hear me and can you see the slides?
Yes, everything's great, Philip. Off you go.
All right, thank you. I'm going to be talking a little bit about digging for treasure in the biomolecular realm. And because I chose this title, I also somehow committed to it. So it's going to have a slight pirate theme to the presentation. I hope you'll enjoy it nevertheless. But I actually start by maybe saying a few words about the group where I work. So the biomolecular chemistry group, we're turning 5 years this year, and we have been set at the Department of Chemistry at the University of Helsinki since the very beginning. And we work with the synthesis and structural elucidation and profiling of different kinds of biomolecules. And you can see some of the snapshots from our recent work here, so very diverse topics. But mostly we focus on application-oriented research set at the interface of chemistry, medicine, and biology. And while we have 2 active projects in the group, so half of the team works with ocular surface research and half of the team with cancer research, I'll today only be presenting on the cancer research, which we are doing. And even more so, I am limiting myself to presenting on a topic that's called boron neutron capture therapy and what kind of a medicinal chemistry project we have going on this very field. We have also been working with other types of cancer treatment systems like antibody drug conjugates, but you can read our work on that on your own. So in line with the pirate theme, so we are going for an adventure to dig for some treasure in the B and CT field. And as you can see here, there is already footsteps from before. So we're obviously not the first group venturing down this road. But also you can see that the export hasn't been found yet. So we are hopeful that we might still locate it before some other teams maybe. But I'll start with a short introduction to boron neutron capture therapy because it's quite a specific field and you might not be familiar with it. The function principle is shown here in the picture. So what you essentially want to do is that you want to develop non-toxic boron delivery agents that are capable of delivering boron-10 atoms to a tumor located, for example, in the head and neck region. Then when you have a good selectivity with boron atoms between the tumor and surrounding tissue, you can employ thermal or epithermal neutron beam. And what happens when you employ this one is that the boron-10 atoms will capture a neutron and undergo a fission reaction that has the destructive power of a single cell. So if you look at this therapy, like from this perspective, it doesn't really matter now if the boron delivery agents are found elsewhere in the body. If they are not toxic, then it's okay. And you can apply this neutron beam in a very narrow region where you would have a tumor present. So you can, in principle, reach selectivity on 2 different fronts. So one, by filling the tumor with boron-10 atoms, and then using the neutron beam in a very selective region. So this is a short introduction to boron neutron capture therapy. If you are interested, there are plenty of good reviews on the topic, which you can read about later on. I'll pick a few of the historical sort of pipeline from this topic. The BNCT torch, it was lit back in 1936 by Locher. And there was a lot of early trials in the United States. But they failed, and they failed because the boron delivery agents used were bad. So people died of the therapy instead of the disease, which is obviously not the place where you want to be. Some light was found in 1958, where Snyder et al. Reported on the synthesis of a boron delivery agent that looks like this. It's called boron phenylalanine, and it has a LAT1 mediated uptake into cancer cells. And it's still today one of the most widely used boron delivery agents in the clinics. The second one that also found its way into clinics and are still in clinical use is sodium borocaptate. It's a boron cluster that looks like this, and it has a passive uptake mechanism into cancer cells, so it diffuses through the cell membrane. I usually ask students on my courses that do you think that a passive transport mechanism is going to be good for reaching selectivity, and it's not good for reaching selectivity. So this agent isn't really good in achieving a selectivity between the tumor and the healthy tissue. And with BPA, the selectivity that you can reach is about 3:1 to 5:1. So it's not optimal, but it works. So that's sort of the baseline here. Also, it's a little bit surprising because when we think about cancer therapies in a wider frame, a lot of progress has obviously happened in the last 60 years. And it's a little bit surprising that in the field of DNCT, the same delivery agents that were developed in the 1950s and 60s are actually still the ones used in the clinics. And today, obviously, as I said, I'm going to be talking about glycoconjugates, and I'm actually going to be talking about the GLUT1 targeting strategy. And so it makes it's worthwhile mentioning also some of the earlier work in the DNCT with glycoconjugates. And the first report you can find in 1988 by Hawthorne's team. They synthesized some glucoconjugates, and there was a lot of work in the late 80s and early 90s on this topic, and then it was a little bit maybe forgotten. And then Titz's group did a lot of amazing work in the field of glycoconjugates for BNCT in the early 2000s. And then there was again a 10-year gap or so, and then the group of Hosman has been doing some work. And again, the torch gets passed between teams, and in 2020, we started publishing our own work on this topic, which I'm going to talk a little bit more about today. So that's some sort of a historical pipeline for you. It's also worth mentioning some of the basic requirements for boron delivery agents. And these are something you can find in the literature, of course. So they have to be able to bring boron atoms to the tumor at a ppm level, so around 20 to 35 micrograms boron 10 atoms per gram of tumor. They need to display minimal systemic toxicity, so they can't really be toxic. And this is especially true for example, BPA, because the dosing used in the clinics is around 400 milligrams per kilogram of patient weight. So if it's toxic compound, it really doesn't work out well. And tumor normal tissue ratios and tumor to blood ratios about 3:1 is what is desirable, but obviously 10:1 would be much better than 3:1. But 3:1 is something that you can reach with BPA, and maybe 2:1 with BSH. And the last thing you need, and this goes true, of course, for all medicinal chemistry related work, is that you really need to have a detailed understanding of the biochemical foundations and also the functional basis of the approach. And in much of the earlier work, this isn't really the case. So you have maybe more to focus on synthesizing the molecules and doing some preliminary studies, but not really hammering out on the biochemical foundations, which are obviously going to be really important in drawing the right conclusions. So when we set out to develop a GLUT1 targeting approach to BNCT, we also thought about potential advantages of such an approach, and of course, potential challenges of such an approach. And this is by no means the full list that you see here. It's just the list that I'm going to have time to maybe go over on today's presentation. But we thought that D-glucose might be actually a really good carrier for boron delivery agents because it's an essential nutrient for all cells. Glucose is also the most abundant biomolecule on Earth, so you're not going to find more biocompatible carriers anywhere. And it also has pre-existing internalization routes to cancer cells via glucose transporters, for example, GLUT1. And we think that you can reach some sort of a selectivity between the tumor cells and healthy tissue based on the Warburg effect, which entails that the glucose metabolism in cancer cells is impaired, and they actually need much more glucose to sustain themselves and proliferate. And many cancer types have been shown to overexpress glucose transporters on the cell membrane in comparison to healthy cells. So we think that this is potential benefits of using a GLUT1 targeting strategy. But there are also challenges that you have to be aware of, and one is the competition with the glucose levels in blood, which are obviously very high, and also the fact that they are high in combination with that the glucose is the molecule that nature has developed over millions of years of evolution for this transporter. So you're going to be facing off against that one, which is not a super easy starting point. And the second one is that you have to be really careful that you don't disrupt metabolic pathways in healthy cells that still sort of use glucose for metabolic energy requirements and such. So there's definitely challenges that need to be solved, and there are also potential benefits that can be used. And since this is an organic chemistry highlight session, to solve or sort of study these factors, you obviously need molecules, and that's sort of where things begin. So you need to be able to synthesize molecules so that you can study them. And today I'll talk a little bit about the first thing that we actually did, and that was synthesizing the positional isomer library of this kind of boron cluster glucoconjugates. We have made quite a lot of this to date. I think over around 40 species of different structure analogs, which is probably the most extensive library, at least for these purposes, ever made. But prior to our work, some of these compounds had also been made. So Hawthorne et al. Early on made, of course, the beta-glycoside, the alpha-glycoside, and then also the conjugate with the boron cluster in position 3. And the reason why these have been made earlier is that the synthetic routes to these are particularly short. So it makes sense for when developing just the synthetic tools to make these kinds of compounds to focus on substrates that you can reach quickly. But when you want to do a medicinal chemistry approach and really understand which position would be worthwhile modifying, then obviously you're going to need all of the compounds to do that. And here you can see some of the synthetic routes we have employed. So some of them are relatively short, so 6-step synthetic routes to end products, some slightly longer, 12-step synthetic routes to end products. And then you can think that if it's 6 to 12 steps per synthetic route, then 40 species, synthesizing them is going to be quite a demanding synthetic project. And that's why I'm really grateful to have talented synthetic chemists in the group, like Jelena Matovic and Jan-Erik Raitanen, who has been contributing to the synthesis of these kinds of compounds. Anyway, synthesis and drawing things, it's of course easy, but you also have to solve the spectra so that you know what you have. And this is something that we focus on in this team, and we use lots of different NMR spectroscopic techniques and also, of course, high resolution mass. So in the case of these compounds, and we have protons and carbons and boron atoms, so we measure proton, carbon, and boron spectra. We do a little of 2D NMR, where we can verify the positions where we have the clusters attached by looking at the HMBC correlations. We do a lot of spectral simulations with the ChemMapper or Purge softwares. So we can see here, for example, the spectra is measured, and you can see here the spectra simulated. And if you just take away the methanol and deuterated methanol signals, you'll see that they look exactly the same. And so by this technique, we actually get the accurate chemical shifts and coupling constants of the compound, so we can deduce also the conformation that this sits in. Something that is, of course, very essential information when you go towards biological applications. But these are generally found in the 4C1 conformations, and yeah, since we have a hemiacetal here, we have mutarotation, so we have both alpha and beta glucopyranoses present. We'll also be solving a lot of other molecule spectra, which are quite difficult, and you can look at our work on polypeptides and lipids in other publications. So it's not only carbohydrates, but also other types of biomolecules. So when we have the library at hand, we decided to start looking at the potential challenges that we had thought about. And the first one is, of course, the competition with D-glucose. And we were in contact with Professor Jarkko Rautio at the University of Eastern Finland and started a collaboration. So we picked a cell line of human origin, the Cal27 cell line. And Jarkko's team first determined the GUT1 expression levels in this cell line. And then they performed a cis inhibition assay using radioactive glucose as the sort of competitor molecule. And then, of course, regular glucose as the control. And what we were really surprised to see is that actually the IC50 values, which in this case indicate how well they bind to the transporter relative to D-glucose, are surprisingly much better for the boron cluster conjugates than for D-glucose as such. So D-glucose takes over 1 millimolar concentration to have a 50% inhibition in the uptake profile of radioactive glucose. But with these, the concentrations are much, much lower. And that's, of course, good. So it means that these kind of cluster conjugates are able to compete for the transporter against the natural substrate. But the follow-up question is, of course, why? And to try and shed some light on this, we started doing some molecular modeling in collaboration with the group of Michael Johansson and also Antti Poso. Michael is at the CSC and Antti is at the University of Eastern Finland. And Iris is working in our team on this topic at the moment. I'm not going to go too much into the detail on the modeling work today. I just want to mention that what we found is that in the sort of transporter and in the carbohydrate recognition domain, where the glucoconjugates are binding, we can see that there is a hydrophobic pocket forming right here on the back, and the boron cluster, which has a hydrophobic nature, fits perfectly well into that pocket. And that kind of explains why we see an affinity increase with these kinds of molecules. Iris also calculated the binding energies, and they were found to be in quite good agreement with the experimental results also obtained. I've been talking a lot about these figures. I think I should also just show what I mean in one figure. So when it comes to competition from the transporter, if we take the glucoconjugates and compare them to D-glucose, this is the situation that we see. So the glycoconjugates, they really are able to compete for the transporter. That's all fine. Then we moved on to the next step, which was sort of trying to understand the metabolic pathway and if there is any disruption to it from the glucoconjugates. And we took a textbook in biochemistry. We looked at the metabolic pathways of D-glucose that look something like this. And then we thought that, well, we might as well just focus on this first step here, which is the conversion of glucose to glucose 6-phosphate. Because if this doesn't take place, then probably we don't have to go on and hammer out all of the different stages of this metabolic route. And the conversion of glucose to glucose 6-phosphate is done by a protein named hexokinase. And so we called a friend, Hannu Mahi, not VTT, and Jelena went over to their facilities to build an NMR spectroscopic assay for evaluating this first step of the sort of metabolic route. So what we essentially did is that we used the preheater block here at the NMR to do an incubation of the different glycoconjugates with human hexokinase 1 that is commercially available. We use a tris buffer containing an excess of ATP, magnesium 2+ ions, and then we monitor the changes in the protein and phosphorus NMR. And with D-glucose, when we sort of benchmarked this protocol, we saw that it got phosphorylated at the 6th position. So ATP was converted to ADP, and we also saw the chemical shift changes in the proton spectra of glucose. And then we tried the same with all of the glucoconjugates, and nothing really happened, which means that they aren't actually recognized as substrates for hexokinase. So they do not get phosphorylated, nor do they enter the metabolic routes of D-glucose. It's still too early to say if they disrupt the metabolic process in some other way, but at least this is a good first step in sort of elucidating this in more detail. With these two studies at hand, we were pretty happy and decided to move forward and look at some essential qualities from maybe a more clinically derived perspective, but still on the in vitro level. And these are toxicity and uptake. And again, we used the Cal27 cell line here for screening of these properties. And we also took now BPA and BSH, the two agents used in the clinics, as comparator or as standards, so that we can compare our results to what is actually used. And so we aren't only comparing here 3 molecules, we are at the same time also comparing a LAT1 targeting strategy, which is the one that boronophenylaniline have. We are also comparing the passive transport strategy, which BSH has, and comparing those to the GLUT1 targeting strategy for molecules. First, we looked at the toxicity profiles, and here we used just BSH as the standard, and that's because it's the more toxic one of the ones used in the clinics. And we can see that the toxicity profiles of the carbohydrates, they come up quite similar to that of BSH, or maybe slightly less toxic, depending a little bit about the concentration. The toxicity studies were done in collaboration with Professor Mirko Sarporanta here at the Department of Chemistry. What we were really surprised to see, though, was when we started looking at the boron delivery capacity, and these uptake studies were actually done also in collaboration with Jarkko Rautio. The first thing we were surprised was about the boron delivery capacity of the molecules actually used in the clinic. So you can see here that they don't really seem to be doing too good of a job. And the second surprise was, of course, to have our own molecules outcompete these on a quite significant level. So in certain concentrations here across the span, concentration range screen, so the glucoconjugate strategy has a 100-fold delivery capacity compared to the molecules in current clinical use. We could also see here that the molecules earlier synthesized by other groups, different anomers of the sugar and the third position, they aren't really behaving that well. So they are better than BPA and BSH, but not close to as good as the optimal positions that we found through this work, which was position 6 in the glucose skeleton. So did we find treasure? I said we went digging for treasure, but did we actually find it? Well, it's too early to say because we still have only published on the in vitro side, and of course we're going to need a lot of in vivo screening to actually say with certainty if this strategy will outcompete the ones in current clinical use. But what I can say is, of course, we are one step closer to properly thanking BPA and BSH for their services to the BNCT community. like you can see from the picture here. And I see that my time is running out, so I'll also be ending. I just want to say that today I showed you a little bit about the groundwork from where we started out on creating a foundation for pursuing a GLUT1 targeting strategy in BNCT. The in vitro picture looks really promising. We can scale the GLUT1 ladder. You'll reach the highest height. In vivo, the picture is still unknown. So we will see what we arrive at as we go. We aren't obviously working alone on this topic. We have a quite large collaboration network and a strong multidisciplinary one set up in Finland especially, and with also parties from other European countries participating, from which we can really take the molecular libraries produced and put them through the entire screening process all the way to preclinical BNCT trials, which is something that isn't found in many places in the world, but in Finland the possibilities are there. I'd also like to thank, of course, the funding agencies for supporting our research in the BNCT field, as well as all other research that we are currently performing, and you for your kind attention. Thank you.
Great, super, Philip. Lots to learn about boron-containing carriers. Thank you very much. Now, there's a question coming in from Carolina Hopia. Many solvents and synthesis reagents aren't something I'd like in my medicine. Do you have to consider residuals dangers at all when thinking of potential applications? Or is it fine since they're removed from the product and screened in clinical studies?
Yeah, well, that is of course true, but you have to understand that at the moment when we are producing things and testing things out, it's one thing. Then if things are promising, always synthetic pathways for making them in the sort of GMP facilities, which is required then for making it for clinical use, is obviously going to be different. And the regulations on what kind of impurities and things you have in your final compounds are really set. So I don't think you would have to worry that something super dangerous would be allowed onto the market for clinical use in this case. The regulations are really, really strict.
Yeah, very good, very good. Thanks, Philip. Again, I can't see any questions coming through from the attendees. One quick question for me, if that's okay. The metabolic stability of those carriers, have you looked into that?
We have looked sort of at the chemical stability, and they are relatively stable in vivo. And of course, it might be a little bit different. There might be some capping of the cluster, for example, creating the nidoform, which is charged. That might happen eventually over time in biological surrounding, but the ether linkage is stable at this position. So we're not expecting to lose the cage completely. Over time, and conversion to the nidoform might also actually help in sort of sustaining it within the intracellular space as it becomes charged and more difficult then to move through the transporter. So might not be a bad thing either.
Yeah, perfect. So maybe just in terms of time, I'll pass over again to Yuri, who's going to introduce the next speaker. Yuri.
Okay, thank you. So our next speaker is Dr. Kaisa Herttunen. She got her PhD in 2012 from the University of Jyväskylä. And after that, she did her postdoctoral in University of Jyväskylä, University of Gothenburg in Sweden, and also in the Institute of Chemical Research of Catalonia, Spain. And after that, she returned to Finland and is currently an Academy of Finland Research Fellow starting from 2019. So please, Kaisa, tell us about your research.
Right. Thank you very much for the kind introduction and invitation to this event. I hope you can see my slides.
Yep, all fine.
All fine. Yeah, let me just go on. So basically today I would like to share with you some stories of host, guest, and self-inclusion complexes, some examples of folding, wrapping, and clipping. So probably many of you are familiar with the concept of supramolecular chemistry that deals with weak intermolecular interactions, and we aim to build highly stable and selective supramolecules or host-guest complexes. And with this, we could aim to efficient separation techniques or analytical applications or sensing. Nature gives us a benchmark in selective anion binding, for example, the phosphate and sulfate binding proteins, where a phosphate binding protein can show a selectivity factor of 10,000 over these two quite similar anions, and 11 hydrogen bonds are forming between the anion and the binding site inside the protein. So there is one guideline how to do more selective and highly binding system is to introduce many bonds, increase the enthalpy. And then there are other factors that sometimes gives us some unexpected results, because also entropy plays an important role. So this is dealing with flexibility and folding processes taking place in our host, and how solvation and desolvation processes affect the stability of our system. The first example I would like to share with you is folding of these oligoamide foldamers that we synthesized, and they have a central 1,3 pyridine 1,3 dicarboxamide group that gives them partial pre-organization, partial control of the shape of the molecule by turning the amide groups, inner amide groups, towards each other. In the solid state, we usually detect that the molecules adopt so-called at conformation, where one of the outer carbonyl groups has turned inside and serves as an acceptor to three hydrogen bonds from the amides. So what happens when we crystallize this system with a small fluoride anion? We observed in the crystal structures that actually now the host is forming a complex where the fluoride is bound with 4 hydrogen bonds from the amides, and also CH anion bonds from the outer aryl rings, forming this really nice helical complex. So we can think of this system as an anion receptor. But what happens in solution? So in acetone, nonpolar or polar non-protic solvent, we can assume that our host is assuming or equilibrating between various conformations. And when we carried out an NMR titration, adding incremental amounts of tetrabutyl ammonium chloride to the solution, we saw gradual shifting of the signals, especially the amide protons A and B, marked with violet and yellow, and also the same ortho aryl proton we detected binding in the crystal structure. So the saturation of the signals was reached at 1 equivalent, and from this we could deduce the stoichiometry, and also that our binding constant is relatively large, over 10,000. From ITC, we got a more accurate value for the binding constant. One way to increase the binding enthalpy is to add electron withdrawing substituents to the terminal aryl groups. So we tested the possibility of adding a cyano substituent, and we saw that indeed the side where we have the substituent shows more strong shifts, downfield shifts in the NMR spectra, indicating a stronger hydrogen bond. However, in the ITC, we were able to see that the binding enthalpy was not changing almost at all, and the binding constant increasement is mainly coming from the entropy term. We carried out some simulations and density functional theory calculations to deduce how replacing the fluoride with the chloride would change the complex, and saw that indeed the structure of the complex is in agreement with our NMR results. And we saw from noisy as well that these protons, the amide protons and the orthoaryl protons, have proximity with each other. One surprise came from when we decided to put more efficient electron withdrawing substituent, this time in the ortho position. And we expected to see higher binding, but instead, this time, the NMR titration curve was showing saturation at 2 equivalents, suggesting that we might have 1 to 2 complex instead. And therefore, when we fitted the NMR data to 1 to 2 binding model with HIP NMR, we saw that the binding constant for the 1-to-1 complex is actually a little bit lower than with the other 2 examples. And based on the NMR spectral shifts, we deduced that perhaps we have this situation that instead of forming the CH anion interactions, this nitro substituent can actually establish anion pi interactions, and this way it would support also 1 to 2 complexation. Second story, I jump into these resocinerine hosts that are macrocyclic supramolecular receptors with an aromatic cavity, and it generally binds cations and electron-rich or electron-poor species. These molecules are known to adopt several conformations with the 3-octal with 8 hydroxyl groups at the upper rim. This ground conformation is stabilized by the intramolecular hydrogen bonding network. And another possible conformation is so-called both, where 2 of the resocinyl rings are pointing up and 2 are horizontally aligned. Over the years, we have used quite often these tetramethoxy derivative, where every second hydroxyl group is protected with a metal and attached crown ethers or other pendant binding sites to it. So this time we prepared a molecule where we have either a paratoluene sulfonyl group or as a reference linker with a metal group here. And from the crystal structure with the tossulate, we saw this quite beautiful and rather rare example that this molecule forms a self-inclusion complex where one of the arms is actually going inside the cavity. So there are 2 different views from the side and from another side to see how the substituent or the group is folding there. But then we were curious to understand if this process is also taking place in solution. So what we can learn about the dynamics of this system. And if we measure an NMR spectra at 30 degrees, what we see is that we have apparent C4V symmetry, indicating that for the vertical and the horizontal signals appear as one, and therefore we most likely assign this as a boat-to-boat exchange process being fast on an NMR timescale, therefore leading to this averaging of the signals. Then we started cooling down the sample and seeing first some broadening and coalescence of the methyl groups at the lower rim, and finally emergence of two sub-signals at -80 degrees. And here what happens is that also now in the aromatic, the horizontal and vertical signals have been separated. For the arm signals that we were curious to see if the arm is going inside or not, we noticed that two of them, ones that assigned to be from the vertical arm, are slightly upfield shifting and broadening. So this suggests that there is another dynamic process going on. And indeed, there might be shielding taking place, indicating that the arm is going in and out. We measured some Roessy spectra, and from this we can see the chemical exchange between the H&T protons and GNS protons on the same phase as the diagonal, and then the coupling through space Roe signals that indicate that indeed the arms that are attached to the horizontal rings are wrapping near the lower rim protons in a similar way as we saw in a crystal structure. We run a conformational search and EFT calculations for this system, and from these results we deduced that most likely one of the arms is included in the cavity, and the arms have several options of wrapping near the cavity. How about if we change the linker from the tosylate to a methylene? In this case, the X-ray structure was showing that we don't have self-inclusion and we don't have any wrapping of the arms. In the NMR at -80 degrees, we saw some upfield shifting of the protons of P that we assigned to the proximity to this aromatic ring shielding effect. And then on the other hand, the N and O protons from the vertical arm were not experiencing upfield shifting. So this likely indicates that we don't have so strong inclusion of the arm in the cavity. So by changing the electronic characteristic of this position clearly affects how this molecule behaves in solution and in the solid state. The third story that I want to tell also involves a third class of supramolecular receptors. These are calyx-4-pyrroles that are known as anion receptors, and these are the most recent results from my group. Calyx-4-pyrroles are synthesized from pyrrole and acetone or other ketone with acid-catalyzed condensation. Since all of these mesopositions are sp3 hybridized, the molecule has a calyx shape. It has several conformations, from which this 1,3-aldehyde is very common for the free host and in nonpolar solvents. And by binding an anion, it can lock the conformation into cone. And now all of the 4 hydrogen bond donors are pointing to the same guest. Then there's some interesting chemistry. We can play with the starting ketone. If we use non-symmetrical one with one aromatic ball, we can create aryl extended calyx for pyrroles, and then if we separate the isomer where all of the aromatic groups are on the same side relative to the cone conformation, we have so-called tetra-alpha isomer. It has this aromatic cavity. And one other option is to strap two opposing mesopositions to increase or generate a more tight binding cavity for anions. So this increases the affinity for chloride, for example. We started studying this parahydroxy phenyl calyx for pyrrole because we are also interested in adding solubilizing groups to this molecule. But in the meanwhile, we discovered that it also has quite interesting host properties as itself, because in principle, it has 3 different binding sites. So we have the anion binding site endo to the cavity. Then we have exobinding cavity on the upper rim. And then when the molecule adopts the cone conformation, we get a cation binding site at the lower rim. And we might think that, okay, the endo binding site is the most prominent for the anion binding, but in this case, we have to remember that sometimes the anion-pi interactions may be repulsive, and therefore a neutral electron-rich guest is a more likely candidate to go inside. In a crystal structure database, we can see many of different solvate structures for this molecule, and it's very common packing feature that these hydroxyl groups generate hydrogen bonded chains throughout the lattice. And we noticed that if we use a suitable ion pair, for example, imidazolium chloride, we can use the chloride as a clip to connect these hydrogen bonded chains. And on the other dimension, we can use imidazolium and to be sandwiched or clipped between the 2 lower rims of the calyx for pyrrole, generating these 2-dimensional sheets. Second calyx for pyrrole example that I have time to present is double strapped calyx for pyrrole, which we bumped into by an accident. Since we were preparing this monostrapped calyx for pyrrole, and in the synthesis pathway, we first prepare this dipyromethane precursor. However, these dipyromethanes are much less stable in acidic conditions than the actual calyx for pyrroles, and therefore they can also undergo so-called scrambling, and then these bonds are broken, and it reverts to the constituent ketone and pyrrole. And this is one probable way that this double-strapped molecule was formed. So basically, this was isolated from the chromatographic fractions of the reaction, and then NMR showed that this has to be a calyx for pyrrole because now the alpha proton is missing. We only see the beta protons, and then the integrals for the strap isophthalo group and the pyrrole signals is giving us a ratio that indicates we need two straps per molecule. From the crystal structure, I saw that the straps are actually this proximally connected, and then we get a nice open structure in the 1,3 alternate conformation. The noisy spectra in the DCM correlated very nicely with the X-ray structure, but when I measured the NMR in DMSO, I saw that the pyrrole NHs were downfield shifting, suggesting that we get a more symmetrical conformation or hydrogen bonding with the solvent. And when we added 3 equivalents of chloride, we saw a slow exchange between the free and bound guest. What's interesting in this case is that typically when we have slow exchange, it means that we have kinetically stable complex that's also giving us high binding constant. But if we need to add 3 equivalents and still there is free host left, thermodynamic stability is not that great. Another thing that we saw is that strap protons are upfield shifting, and especially, with the benzoate, the bound guest gets clipped or squeezed between the straps in this condition. So to get some idea of the energy barrier, we ran some noisy NMR and used EuChemS software to compare that with this system with 1 strap and known high affinity 10 to the 5 molar for chloride. We get energy barrier for the complex dissociation around 17 kilocalories per mole, and with our double strap system, the energy barrier is almost in similar magnitude. So even though we cannot make thermodynamically very stable complex, it's still kinetically stabilized. And with some imagination, I compare this system to a Venus flytrap that can capture its guest, rather reluctant guest within the petals. I think my time is running out, so I wish to conclude by summarizing that we can control the folding and conformations of our systems with anions, ion pairs, and with structural functionalization of the host molecules. And finally, I want to acknowledge all the co-authors and collaborators behind this work and my current group that is being funded by the Academy of Finland. And thank you for your attention.
Super. Thank you very much, Kaisa. And there's a question from Jan Deska. Jan, you can ask away.
All right. Yeah. Thank you very much, Kaisa. For your first, can you hear me?
Yes.
Great. Okay. For your the first project, have you considered to look into anion binding catalysis with those chloride or fluoride binders? They give a very nice helical shape, and I've seen similar systems where they are used to activate carbon-chlorine bonds or maybe even cooler carbon-fluorine bonds. Have you considered this?
I haven't considered that, but we have a joint project going on with Petri Pihko that involves this kind of catalysis. So let's see what the future brings. Thank you for the suggestion.
Thank you.
Okay, thank you. So our next speaker is Professor Jan Deska. He got his PhD in 2007 from the Saarland University, German. And after that, he has worked in several universities, both in Germany and Sweden, before moving to Finland. He started in Finland in 2015 at Aalto University, and currently he is working as a professor of organic chemistry in the University of Finland, University of Helsinki. So please, Jan, tell about your interesting topic.
Yeah, thank you very much, Yuri. Yeah, I want to start with thank you, Pat, and the EuChemS for setting up those national highlight sessions. It's a great thing, and special thanks to Yuri and Ari to actually get our Finnish session off the ground. I want to get back to what Juha was starting in the beginning, that we take inspiration from nature, especially in Finland, but I think quite generally chemists look for inspiration in nature when designing chemical processes, when designing, for example, catalysis principles. So we look into active sites of enzymes to understand how nature is developing catalysts, for example, for carbon-carbon bond formation. And by understanding the catalytic principles, we can then come up with our small molecule analogs that may precede carbon-carbon bond formation in a similar way. And along the same lines, we can look into nature in trying to understand how nature constructs complex building blocks. We can look at the natural strategy and how molecules should be functionalized, how molecules should be aligned to then snap into a more complex structure, such as an sterol synthesis. And we can mimic that with chemical means. Having said that, only few chemists actually use natural tools in their everyday life. So we see rather rarely enzymes or nature being involved in the production of chemicals. And there's of course the bright side of biological catalysts, of enzymes especially. So there are examples up to the industrial scale where we see enzymatic systems being used for the preparation of chiral building blocks, alcohols, amines, amino acids. And enzyme catalysis provides, of course, very benign conditions. No hazardous solvents, mild reagents. And with modern biotechnology, we are able to fine-tune our biological catalysts in a similar way as we would fine-tune our ligand system along a metal center in transition metal catalysis. Another great benefit of biological catalysts is that they can be combined with other enzymes. Nature uses a cellular system where thousands of different proteins swim around, and they can act on their designated reaction without interference by the others. So a cascade design seems to be more plausible with a biological catalyst. One of the reasons why we don't see enzymes too often in synthetic chemistry is that we as synthetic chemists have to rely on all reactions available. We cannot, or rarely, really choose our reaction pathway, our synthesis strategy based on what's available. And that's the problem. Nature doesn't have many reactions available. There are few privileged enzymatic transformations that are relevant. There are much more reactions found. We know they exist, but we might not be able to actually use those enzymes yet. And then there's a whole lot of reactions that is just not found in nature, that's not found in biosynthesis. So we would assume that we can't use those reactions for synthetic purposes, or those enzymes. A bit of a different picture of where I want to show the entirety of reactions that we find in biology, in biosynthesis. And on the right-hand side, we see the entirety of reactions that modern organic synthesis has been developing over the last 200 years. And there's a good overlap, but we can see there is a lot missing, and there is for different reasons. There are a lot of reactions that are not used by nature in the biosynthesis. So our questions, or one of our research questions in my group, is that, can we convince nature to do those reactions? And this brings us to a topic that has been booming over the last 10 years, that I like to quote term reverse biomedics, where we use modern synthetic organic chemistry as a role model for natural catalysts. So we are not bio-inspired, we're actually chemo-inspired. We're creating biological systems that are inspired by chemistry. And probably the most prominent example is the transformation of an oxygenating enzyme, cytochrome, to a carbene transfer biocatalyst in the group of Francis Arnold. And that whole heme biocatalyst transformation scene has been really booming over, as I said, over the last decade. So we've seen all kinds of activations to unlock novel reactivities, CH activation chemistries, nitrogen transfer. We've seen photo redox-based transformations where enzymes are not used in the ground state, but actually photo-excited to access new reactivities. And we see de novo protein engineering, where, based on computer models and the design of new proteins, we can access new reactivities. Now, as a chemist, we might ask, sure, but why would you do that? We do have really good catalysts. We do have great selections of transition metal catalysts for all kinds of reactions. We do have the field of organocatalysis that is giving great opportunities, and all the other ways of catalyzing reactions. Now, my argument would be that yes, so we start with a key chemical transformation that we would like to catalyze, and we start mining activities and evolve our enzymes to access those novel enzymatic activities. And those enzymes might be as good as the chemical blueprint. They might be not as good, but they offer something more. As I said before, they offer the possibility to combine those novel activities with existing activities and create enzyme cascades for one-pot designs. Or, knowing the protein structure, we can now genetically encode those proteins into microbial organisms and microbial hosts. So we can generate our microbes to engage in this kind of catalysis and this kind of chemistry. And we'll start with a quick story that I've been talking about for a while now, and that is looking at natural product synthesis using natural systems, but not using biosynthesis, but actually looking at the chemical blueprint. So how do chemists synthesize natural products? And we've chosen Angiopterlacton B as an interesting target here. Moderately complex, a few stereogenic centers, probably biosynthetically linked to osmundarlactone, which is already a much smaller molecule, much easier to understand. And while we've been working there, we've seen that also chemists have been interested in that molecule, synthetic chemists that do not have the biocatalysis background, started to work on that. And so in 2017, we've seen this very streamlined and elegant synthesis by Lawrence to access angiopterlactone based on 4 reactions: an asymmetric reduction, an Akhmatovich rearrangement, a redox isomerization, and a base-induced dimerization. And we thought this is quite interesting. That's moderately complex in number of steps. So you could actually design a biosynthetic machinery based on that. So as you know from biosynthesis, we have all those blocks with abbreviations that stand for enzymes, and we can show the entire metabolism of organisms based on those systems. So we could design our really small biosynthetic machinery to mimic the classical retrosynthetic analysis. So what we would need a reductase, an Akhmatovich oxygenase, an isomerase, and something that stitches together those 2 molecules in the dimerization, plus 2 or more auxiliary enzymes. This sounds all good until you realize that, well, there are at least 3 out of those 6 modules that are speculative in function. You don't find them usually in biosynthesis. And so we've been developing tools, enzymatic tools that actually, allow conducting those transformations based on a protein-based scaffold. So we developed a method for the Akhmatovich ring expansion of furans, and we developed an enzymatic method for the isomerization of those Akhmatovich pyrenones to the corresponding lactones. So we could make, for example, valtrolactone, but we could also make the osmundarlactone based on the multi-enzyme one-pot enzyme cascade. And then we were lucky and actually found that just adapting the Lawrence protocol ever so slightly, we can actually run that dimerization in water. So overall, we have a fully water-based system to access the rather complex tricyclic natural product. And as I said before, the other big thing that we can do with biocatalytic systems is that we can take the proteins that we identified to engage in the non-natural chemistry. So let's look again at the Akhmatovich module and the boron hydrogen module. So we can take those. Enzymes. We can genetically encode them into E. coli, into bacterium, and now we have a protein and have a living system that now does the non-natural chemistry for us. But yeah, let me take one step back. This is the organic chemistry session, so I will talk less about the biology and focus on synthesis. And today I want to show you a little bit of a recent project that we've been engaged that was looking at biocatalytic oxidative CN bond formation. So showing a little bit how we work out those non-natural modules. Where do we start from? What is our inspiration, and how do we proceed? And yeah, I guess we're all organic chemists, so I don't have to tell much about the importance of carbon-nitrogen bonds. Biocatalysis has been quite a good tool over the last years with the advent of transaminases and imine reductases. They are really potent tools to introduce nitrogen functionalities into molecules. However, always as a functional group interconversion, where you take a carbonyl functionalized molecule and catalyze a reductive amination to introduce the nitrogen to the molecule. So we were more interested into oxidative CN bond formations. And again, look into the chemistry. How do you do that? How do you activate less activated molecules that are not carbonyl containing, or where the carbonyl is not the crucial point? So in best case, sp3 carbon hydrogen activation. And there are, of course, examples, usually with some kind of activation in terms of olefins. We seen that Wacker type chemistry of white to oxidatively couple carbon and nitrogen, which is biochemically challenging. That goes via palladium, allyl palladium complexes, which are a bit tricky to mimic in a natural setting. Or we can look into nitrine insertion chemistry. In this case, the sulfone amide is oxidatively activated. A rhodium nitrine is formed that now inserts into the carbon-hydrogen bond. In that case, actually, Arnold has also picked up that kind of chemistry and developed by directed evolution an enzyme that can engage in a similar transformation. Now, we were interested in a thing called the Ene reaction, or aldehyde reaction. And an aldehyde reaction where our ene would actually consist of two heteroatoms, a nitrogen and oxygen, so-called nitroso compound. And yeah, so there's ample precedence in literature about functionalization of allylic compounds using those acyl nitroso species as intermediates. They're always formed in some way. They're not really stable, but you can form them catalytically, for example, by oxidation of hydroxamic acids. And then in this ene-type reaction, a new carbon-carbon bond is formed by activation of the allylic CH bond. And yeah, just the similarities between the Diels-Alder cycloaddition and the aldehyde coupling reaction that we will come back to in a minute. Especially if you look into how those nitroso compounds are formed, they are formed from hydroxamic acids or related compounds. And those hydroxamic acids or hydroxylamines, they're actually used quite frequently in biocatalysis, not for CN bond formation, but as redox mediators. And we know that there are enzymes that can convert hydroxamic acids into O radicals to do single electron transfer reactions, or that can oxidize tempol to the oxoammonium species in 2-electron oxidations, for example, alkyl oxidations. So we thought if enzymes can activate those kind of molecules, maybe they could also engage in oxidation of those hydroxamic acids. Another model substrate was this, well, not a hydroxamic acid, an N-hydroxy carbamate with a suitable allylic functionality in the side chain. And we've been screening lactases, which are blue copper enzymes. They're quite commonly used in those tempole oxoammonium dehydrogenations. So we thought that there might be something in that. And indeed there is. So we did identify 2 lactases that give really high conversions, up to full conversions of our model substrate, and where we can also see moderate to good yields and selectivity for the desired carbon-nitrogen bond formed the heterocycle. Looking back at our Akhmatovich chemistry, we've been dealing with peroxidases for a really long time, so we thought maybe let's also get back to the peroxidases. And interestingly, horseradish peroxidase, which I would consider as quite a dull enzyme usually, is actually excellent for this transformation, giving full conversions and almost perfect selectivities for the product. So we have at least 2 very good candidates for the further development of the method. And so we started to look into substrate scope. We've initially been focusing on the horseradish peroxidase, which gives generally good to very good yields for most transformations. So we see all kinds of architectures being tolerated. So we can use the N-hydroxy carbamates as well as the hydroxamic acids. We can make spiral compounds. We can make 6-membered ring systems and so on. And for the substrates that we tested with the lactase, the copper enzyme, we do see also formation of the product, not always with those high yields, but still quite a decent catalyst. Now the question is, what is going on here? So we have 2 systems. How are they similar? Where do they differ? Is it an enzyme-induced reaction, enzyme-assisted, enzyme-catalyzed? And so we've been having some discussions, I would say, in the publication process. It's not yet published, but especially bringing up this question. So what do you know about the chemistry at the enzyme? Does the enzyme catalyze the carbon-nitrogen bond formation? Does it maybe only catalyze the formation of the nitroso compound? Does it diffuse out before it cyclizes? Or is there some kind of intimate contact between the metal center, copper in case of lactase, or iron in case of the peroxidase, and the substrate? Other questions: is it a radical process where there's a 2-electron process, and so on? So we started to look a little bit into the kinetics and stereochemistry of the reactions. So if we label our model substrate with 3 deuterium atoms at 1 methyl group, we see that the CH bond cleavage proceeds selectively trans to the functionalization, which is actually in line with the intermediate that has been discussed previously, where the nitroso compound is attacked by the CC double bond for this aziridinium oxide intermediate. Similarly, if we totally deuterate our substrate on the methyl groups, we can now determine kinetic isotope effects. And for the horseradish peroxidase, we see an inverse secondary isotope effect that is much less pronounced for the lactase. But in both cases, that would again somehow indicate that we have an aziridinium oxide intermediate involved. Interestingly, if we look into enhancer selectivity, we've seen that the horseradish peroxidase, whatever we try to do, gives us racemic product all the time. However, when we go for the lacase, for the lacases, for the copper enzymes, we do see minimal enantiomer induction under neutral conditions, and if we get slightly more acidic, which you do see up to 40% EE in the product formation. That's not great, but on the other hand, lacases are not known to be great, give great enantiomer induction. They actually usually give racemic products. What we take from that is that most likely for peroxidases, we do have diffusion of the nitroso species, while in the lacase case, we do have a true biocatalyst where the substrate, the oxidized substrate, has to be somehow still bound to one of the copper centers. And yeah, a little bit on the extension of the scope. So we looked into bimolecular couplings, so we can get the similar nitroso-ene chemistry also with small enes, small olefins that are not attached to the hydroxy carbamate. We can extend the whole thing into Diels-Alder chemistry, which again pinpoints, or is for us a good indication that we do deal with nitroso compounds and not some radical species. And we sometimes see some weird reactivity, in this case, the amido hydroxylation. Then expanded, looked at the broader scope. So yes, the intermolecular couplings can be done rather general. Not all substrates work equally well, but you get your reaction running. Same goes for Diels-Alder reactions, replacing the allylic olefin by a diene, by 1,3-diene. We can see under similar conditions also Diels-Alder cycloadditions proceeding. So yeah, this brings me to the end already. So quickly summarizing, what is the status of our enzymatic nitroso chemistry? I think both systems provide very mild conditions. They both use air as terminal oxidant. And overall, we see quite a broad reaction scope, broad substrate scope, and have a very nice access to complex heterocyclic building blocks. In case of lacase, we now have a very rare example for enantiomer selectivity for this special enzyme class, which of course makes it a really attractive target for protein engineering. And again, since we are enzymatic, we can combine different enzymes, we can combine different transformations. So we can, for example, combine deformation of a hydroxamic acid, lipase catalyzed. You take a simple ester and hydroxylamine, and you can then use a lipase to catalyze the formation of the hydroxamic acid, and you combine that with the nitroso-ene-ase biocatalysis to get more complex building blocks, get the CN bond formation. Yeah, with that, I'm at the end of my talk. I hope I could give you a comprehensive overview of what we're doing. So we're looking for artificial enzyme functions. We try to use those in chemoenzymatic natural product synthesis. And we try to bring the artificial enzyme functions into nature and make tailor-made cellular factories. We also look a lot into chemoenzymatics in terms of combining metal catalysis and enzyme catalysis. And yeah, with that, I'm at the end. Of course, I have to thank my group. I've already indicated the people who did the work. And I want to thank all the funding agencies that provide us with the money to do what we like. And yeah, thank you for your kind attention.
Great, super, Jan. We've already got some questions coming in through the Q&A. Actually, one from my own PhD student, Lucy Byrne. Thank you for your talk. Sorry, my microphone doesn't work. Do you know why lacase gives enantioselectivity and other enzymes would not? Can you try other enzymes with similar active sites to increase the EE? Thank you.
Yes. So we looked a little bit into the structure of the lacase, and we see also quite some differences in enantiomer induction for different lacases. We're at the moment starting to use docking to actually see if there is a. First, we thought it would be the copper center, the T1 copper of the lacase, that would be the Lewis acid to bind the nitroso compound, and there are crystal structures of copper nitroso compounds, so that there can be an induction. we now actually think there might be something with amino acids close to the active site that might provide hydrogen bonding interaction with the acyl nitroso compound. But at the moment, yeah, we can't really tell yet what that is. We just go into docking where we have different laccases that differ very strongly in EE. We've tried a few other copper things because it's superoxide dismutase that, as far as I remember, didn't give any EE, which is again structurally rather different from the laccase in the active site.
Great, thanks for that. And a comment from Ilan Marek. Very nice talk, Jan. Warmest congrats. So there's from the Q&A. I'm just checking any of the attendees have a question that they'd like to use the raise hand function. See any other questions coming through. Of course, for years, people search for a Diels-Alder A's. So it's interesting to see EuChemo inspired to actually start looking, not looking for them, but actually just bringing the chemistry to the enzyme and see what happens. That's really an interesting approach. Now, I don't see any other further questions. And we're coming up smack on 3 o'clock. So maybe we leave it there, Jan, in terms of the interest of time. So again, thanks very much. And I'll pass over to Yuri for the next introduction.
Thank you. So it was pretty interesting talk, actually. I think I should have read more about biochemistry to understand that more, but it was interesting. Our next speaker will be Professor Petri Pihko. He got his PhD in 1999 from the Helsinki University of Technology, which is nowadays known as Aalto University. And after his PhD, Petri carried out his postdoctoral research in the Scripps Research Institute at California, moved back to Finland, and after a couple of years at Helsinki, moved to University of Jyväskylä at 2008. And currently he's working as a professor of organic chemistry there. So Petri, please tell about your research.
Thank you, Yuri, and I would also join others in thanking the organizers for organizing this fantastic event. So I would like to, I have read this rather provocative title, and it's about, I'm actually going to continue a little bit in the same theme than that was introduced by Jan, but also by Kai. So this is a kind of a hybrid on, well, not enzyme catalysts, but maybe enzyme mimetics, and then also maybe some supramolecular chemistry as well. So I will talk about two catalyst families, which may look like that we designed them, but I would like to maybe challenge this view that did we actually design them, or did we arrive at them somehow by luck or by some other means. So how would you design catalysts? Let me go back to more than 100 years ago. A very pre-science chemist, Wilhelm Ostwald, who also received the Nobel Prize in 1901, had this vision that he thought that there will be some transitional forms between enzymes and then organic compounds. So this is much before there were any thoughts on organocatalysis, something which already arose in the 20s and arose as a concept in the early 20s and 30s in Germany, and then of course it has been popularized and expanded much later by the last year's Nobel Prize, Ben List and Dave McMillan. But Ostwald was, I think, way ahead of his time. He was thinking that we can find compounds that are somehow between enzymes and then between and then organic compounds. So This my view of organocatalysis is that this is what organocatalysts are, that they are these transitional forms. And in fact, I would like to say that at least in the case of the work of Ben List and also the late Carlos Barbas, the very early work was very much inspired by natural catalysts, not only by enzymes, but also by catalytic antibodies. So we owe a lot to these wonderful catalysts. However, we synthetic chemists, we would like to make something which enzymes cannot make. We would like to make natural products through other means. So where do we need these catalysts? Well, one example would be complex natural products of this type. So as you can see, there are lots of different stereogenic centers in these compounds. Many of them contain a methyl substituent, and these are everywhere. This is actually fairly common because this is derived from the methyl malonyl unit in their biosynthesis. So can we design a catalyst that makes these systems, so using, for instance, a reaction something that looks something like this? So here we have a nucleophilic partner here, and then we have an electrophilic partner. Now the trick is that here we actually would need selectivity on this nucleophilic partner. We would like to be able to control the phase selectivity here. However, what's easier to activate is this electrophilic partner. This is something that, thanks to the work of Dave Macmillan and co-workers, we know how to do that using iminium catalysis. But in their systems, you always had selectivity on the phase of the electrophile, or sometimes a combination, but never only on the nucleophile. So how do you design something like this? Well, we had some early success with this catalyst, this diphenylpyrrolidine catalyst that was earlier used by Carl Anker Jørgensen and his group for something completely different. We just happened to have it in-house. So we used it for this reaction, and it actually gave fantastic selectivity. We couldn't understand how, but then computations by our collaborators suggested that dispersion interactions are important. Some of these are highlighted in these green areas here in this figure. So these green areas represent attractive dispersion interactions in one of the transition states that leads to product. This is actually a rather complex story, but just to give you some sort of rule of thumb, the contact between this iminium ion here and then this nucleophile, this is what's important also for ensuring the selectivity. Now, if this is true, then this suggests that actually it's not that easy to discover a catalyst where you would be able to use a smaller system, something like this, as we originally planned, because the contact area will be smaller. But we decided to try it out because in theory, something like this should be possible. So how do you do this? Well, we started out with this simple pyrrolidine catalyst. We also tried the Macmillan catalyst. We tried the Jørgensen type catalyst, other Jørgensen type catalysts. We tried many different catalysts. We didn't seem to be able to go anywhere. With some of them, we got reasonable selectivities. Some of them gave no reaction or no selectivity. Then we decided to make some changes to this so that we would be able to arrive at a more modular structure, something which can be modified more easily. So we started from pyroglutamic acid, and according to the precedent by the Trost group and others, we basically made compounds where we had an aromatic group here, and then here we could have something that you could make from this ester group, let's say tertiary alcohol, for instance. So this gives us a a more modular catalyst which you can optimize more systematically. So first we check the effect, the easiest thing to do. We check the effect of the silyl group. This didn't work. There was no improvement. So these the figures here, the last figure is the selectivity. The first figure tells you basically how far the reaction goes, because there are also some side products, some hydrolysis products. So maybe you just take a look at the second figure because it's more important. So then we try to optimize the R group here, and there was almost no effect. It just turns out that electron deficient groups were detrimental to selectivity. So we better keep it electron rich. Actually, there was a slight increase in selectivity with slightly more electron rich groups here. So finally, the most challenging, of course, to optimize, but the most important was this other group here. And already the French postdoc who did this work did a fantastic job on finding out that, in fact, if you also put an electron donating group here, and especially if you also put some metal groups, therapeutic groups are too bulky, but if you put some metal groups, you actually end up with a catalyst that seems to be promising already. So when we combine all of these things, we have the optimized silyl group, we have the optimized for this Western aryl group, and then we include the optimal Eastern aryl group. We could already predict that by combining all of this, we would probably arrive at the winner catalyst. And indeed we did. So with this combination, you have the best selectivity and also the best conversion to the product. This is a very interesting system because actually it involves substituents on both Eastern and Western sides. So you can actually do kind of a double Hammett. So we actually came up with this equation that describes that the Western half is basically has a larger influence compared to the Eastern half. And there's some correlation. The correlation is not great, but anyway, you will see that the worst catalysts are over there, and the best, and then the best catalysts are here. This 12N has reasonable selectivity in this system. I know that some people might ask the question that can you rationalize why this is? And we actually tried. We tried to go both computationally. We also have tried to make a catalyst where we have something different here in the Western side, including the indole moiety. We still don't know. I'm sorry, we still don't know why this is a good catalyst. We can speculate that maybe this is a good catalyst because some of these more electron poor catalysts induce a side reaction, but we actually haven't been able to come up with definite evidence for that either. However, just because we have a good catalyst, let's see how it can be used, and we can use it for many things. So it's not restricted to this methyl group. We can also put different alkyl groups here. And it's not so surprising that if you put a more bulky group, you get better selectivity also. In addition, these methyl building blocks, you can make, for instance, chiral metal containing aldehydes. You can make chiral metal containing ketones, and you can even use it in natural product synthesis. So, for instance, we made in 5 steps this segment of bistramid A, which previously has been made in approximately 10 steps or so. So this is a fantastic catalyst system, which we are also currently using for other total synthesis projects. Hopefully we can communicate them soon. I just would like to continue with this, that we also have used the same theme, this Mukadam and Michael team for other families. Maybe the most interesting case is this lycoperiodic acid, where we actually tried to use something very similar compared to the original system. So we also used the siloxafuran, but we just used an ester substitute siloxafuran. So in this case, it turns out that the previous winner catalyst gave no selectivity, and also the previous winner with simple siloxafuran also gave more or less no selectivity. We used this catalyst for the total synthesis, and then Sami recorded in the final days of his thesis, he dug out some of the old catalysts that Orally had left behind and found out that in fact this was even slightly better. This is a rather sobering lesson, actually. Does it mean that we need to make a new catalyst each time when we have a new problem? We are not sure, but this is it's not easy to design a universal catalyst. This is actually a pressing problem in organic chemistry at present. So one way that we thought that maybe we could approach this problem in parallel, we have been looking at catalysts that are maybe a little bit more enzyme-like. So we thought that it should maybe have, maybe it should be bifunctional, something like this, but not as simple as this, and neither as simple as this, but something that actually folds. So we were inspired by the work of Martin Smith and co-workers at the University of Oxford, who published their monofunctional catalyst in Angewandte more than 10 years ago. We were inspired by that work. We later found that actually they had also made the bifunctional catalyst as well, but we were not aware of that at the time when we started this. So anyway, this is a system that folds. Where we would intend to use it? Well, basically, we would like to use it for something like this, for catalytic Mannich reaction. That was a problem that was essentially solved, except that there was no good solution for aliphatic imines, which would be actually, and these imines and these products actually would be quite useful for total synthesis of natural products if you can actually use aliphatic imines instead of just only aromatic imines, which was kind of a solved problem. So we tried some of the simpler catalysts with these aliphatic imines, and indeed they do not work. There's some apparently some either product inhibition or then just decomposition, but this doesn't go anywhere. But even with aromatic imines, our first design didn't work either. And we found out why. It has a rather sterically crowded active site. So the design by the Smith group had to be modified. So we modified it basically by replacing the linker, by using this amino-indole linker. Now, it may sound like we designed this very soon. We thought we were very smart when we designed this. Actually, the truth is that we had just happened to have this amino-indole in-house from another project. And then this another French postdoc, Nicolas Probst, he basically just used it because it was available. And we then later found that this was a very good lucky accident, because now we got a more open and accessible active site that still maintained this intramolecular hydrogen bond. It maintained the fold, but now it was more accessible. And this catalyst was indeed much better. So not only did it work with these aromatic imines, but it worked very, very nicely with these aliphatic imines, giving very good yields and selectivities. We had to use in excess of this aliphatic imine, but this was not a big problem. Typically only twofold excess was sufficient. And it also worked at a lower temperature. So at 1 mole% concentration, it worked very nicely. You can do it in a gram scale reaction. And you could easily access the opposite enantiomers simply by switching this the chirality of this end unit. And you don't need to go to very low temperatures. 0 degrees is enough for many of these reactions. And we also expanded this scope of the methodology to beta ketoesters as well. So instead of using this simple dimethyl amino catalyst, a new catalyst, which actually was even tighter, included this piperidine moiety, and this was a much better catalyst for these. which had basically a good substrate scope for both beta ketoesters as well as these malonates. So how does this catalyst work? So we think it works by binding both substrates, and then there's a product release step. And we have actually tried to understand this from many angles for several reasons. One of the reasons is that this is, well, it's a bit like enzymes, so it works wonderfully for a manic reaction. And then some people have asked, okay, can you send this catalyst to us? Can we use it for something else? And then we have sent it over. And then people report, yeah, it works, but it gives rather poor selectivity or poor conversion. So it's very picky. It's very selective for this reaction. So why that is, it looks like that again, this is with the help of our collaborators. It looks like that it actually binds both substrates in a rather tight transition state, where the substrates are contacting the pocket in the catalyst. And this is maybe best described in a comparison of the transition of the computed transition states for our own catalyst and then the Takemoto catalyst, which doesn't have this catalytic pocket. So with aromatic amines, both catalysts do work. With aliphatic amines, the transition state with Takemoto catalyst has higher energy, but actually the reverse is true. I don't know if these absolute numbers are probably not correct, but the trend is clear that in fact our catalyst can accept aliphatic substrates much better, because apparently it has these attractive dispersion interactions. So instead of just presenting these active site functionalities in the correct positions, we here have a proper binding site, which is a little bit more similar already to how enzymes catalyze reactions. So the overall fold is important, but this kind of folding is difficult to design, and we have been presented with many surprises here. One of the surprises is this. So actually this catalyst can adopt two different modes. So there's this so-called cooperative mode, which is desired, where we have a urea forming a hydrogen bond with the thiourea. And then we have a kind of a pocket. This is not a malonate, this is HF agar, but this is a, well, surrogate for the malonate. And then we have an alternative mode. This was again discovered by accident, but then we have later found that this is actually a very, very common feature. So whenever you put a very small anion, be it fluoride, chloride, bromide, we haven't tried iodide, but it probably works the same. And without regard to the stereochemistry, it always refolds. The thiourea flips over, it refolds and forms this system. And in solution, it does the same. So this test was an X-ray structure, but in solution, it does basically the same thing. So it's maintained in solution. And well, when you add the chloride, it changes its shape, and this is a dynamic system. So if you add to this catalyst, if you have tetrabutyl ammonium chloride, it more or less completely changes its shape to this. So is this something that you can design, or this is something that you can just well discover by accident? We think this is a very exciting system because it actually resembles another feature of enzymes, which is called allostery and allosteric regulation. So in enzymes, very often you can have phosphate and sometimes also chlorides, and then you can bind these anions and they change the shape. So for regulating proteins, and there are also enzymes where chloride is an important activator, for instance, in amylase, which also in human amylase, this is also activated by chloride. So we think this is a very interesting system, and if I would have a lot more time, we could also talk a lot more about how this would work. But this is still an ongoing work where we are trying to understand the system. So we have found basically that chloride inhibits these reactions, again by changing the shape of the molecule, and we have also found that this can be done under a variety of conditions, and this can probably lead to very exciting science in the future. But I think this brings me to the end of this talk. So I tried to ask a question: can we really design catalysts? Can we design catalysts that fold and work like enzymes? Maybe, but I think you also have to admit that, or at least I would like to admit that we have also been lucky. We were lucky because we were able to get structures from these many of these complexes just because they happen to crystallize, just because in Jyväskylä we have one of the best X-ray facilities of the Nordic countries, and we have people who know to use them. So you have to get this lucky accident also in your research in order to push you forward. So I would like to acknowledge several people who contributed this work. I hope I don't miss anybody, but including Aurelie, the chief postdoc behind this, Nicholas, and many others, Antti and Dimitris, also our collaborators in Hungary and also in Jyväskylä for DFT and X-ray help, and then of course our funding agencies. This is what we are, and if you want to take a further look, closer look what we do, please visit our website. Thanks a lot.
Super, Petri. Very nice talk. Very elegant work. Well done. Again, it's open for questions from the audience. You can either use the Q&A box at the bottom of the webinar thing, or just raise your hand in the normal Zoom way, and then I'll let you speak to ask the question. Maybe while we're waiting for the audience to come up with a question, Petri, just in terms of the work later on where you chanced upon the indinol unit as being key, and of course there's 2 chiral centers there, and then on your cyclohexyldiamine you got another 2 chiral centers there. Did you find many examples of mismatch? Did it take a while to figure out which was the right set, or did it change for different transformations?
Maybe I will return to the catalyst design right here. So it actually turns out that somehow the key to the enantiomer selectivity is this amine moiety here, and the indinol has rather minor effect. So there's some mismatch, so that it turns out that this is actually a better pair, something which actually agrees on the finding of the Smith group, but both do work.
Right.
So this is a little bit, it's a bit sad actually, because this is not, we could probably replace this with an achiral system. It's just not easy to find a system which has the same rigidity.
Yes. Very good, very good. Again, there's a question coming through from Ilan Marek, I should say. What about more substituted malonate to reach quaternary stereocenters?
We tried. They don't work so well, but we actually haven't given them a more systematic attempt. This would be, we should probably take another look now that we have more catalysts. So
yeah, because formation of quaternary stereocenters is a tough deal, to put it mildly, but your system
it is. Yeah,
looks well primed to be able to do that.
Should take another look.
Very good. And there's another question from Eugenius. Hi, Eugenius. Petri, a very nice talk. Sometimes there is coincidence of factors, but actually you had an idea which finally afforded a wonderful result. Do you have an idea of modeling such accidents? Now, that's a philosophical question for a Friday afternoon.
Yeah, I don't have an idea of modeling, but what I would recommend is I think we should always also do the experiment that we don't think that should work. So this controlled experiment and then just that let's just try it out anyway. Experiments, these are very important.
Yep, during the final year of my PhD with Sir Derek Barton, he always, if I ever asked a question, his answer was always, well, just do the reaction.
Yes.
Good advice. Nike obviously took that up as their slogan later on, and just do it. Again, any more questions coming through from the chat? Doesn't appear to be, or none coming through from raised hands either. If that's the case, Petri, thanks again. Super presentation. Good to see you again.
Thank you. Good to see you as well.
Yeah. Over to you, Yuri.
Thank you. And then we have the next speaker and also our last speaker today. So Dr. Toni Metsänen. So he got his PhD in 2016 from the Technical University of Berlin, Germany. And after that, he moved to USA to carry out his postdoc in the University of Utah and returned to Finland after a couple of years. After working also in the University of Jyväskylä, Toni moved to Orion Pharma to work as a senior scientist. So please, Toni, tell us about your research.
Great, thank you, Yuri, for the kind introduction, and thank you, Ari and Yuri, for organizing this event. It's been really interesting to see all kinds of chemistry that's been conducted across Finland, and obviously it's an honor to be part of the program here as well, representing the industrial side of the organic chemistry in Finland. So as you mentioned, my name is Toni Metsänen. I'm a senior scientist at Orion Pharma, and my business at Orion is to be a scientist in the medical chemistry department. And what I'll present to you today is ongoing collaboration we have currently with the University of Helsinki. And before going to the actual science, I want to make sure that I highlight all the people involved in this, because it's obviously not something I'm working on my own. Let's see there. So we have this collaboration between University of Helsinki and Orion Pharma, and from the University of Helsinki side, we have Professor Jari Yli-Kauhaluoma from Faculty of Pharmacy, and he's actually the principal investigator on this project. And then from the Department of Chemistry, we have Professor Timo Repo, who's technically an inorganic chemist, but some of you in the audience might recognize him from the frustrated Lewis pair chemistry. That's him. And then we have from the Department of Computer Science, Professor Arto Klami. He's really the machine learning or artificial intelligence, however you want to call it, expert in this project. And then from Orion, we have myself, and then Dr. Julius Sipilä from the Digital Medicine Department. And finally, most importantly, to make this possible, we have generous funding from Vaikuttavuussäätiö. This is the Finnish Research Impact Foundation. And with their funding, we've been able to hire a postdoctoral fellow, Juri Timonen, that you just heard there, to actually do all the work on this project. So Juri's been now half a year with us in Orion, and now he's currently moving to University of Helsinki, and he'll spend a year there before returning back for another half a year at Orion. And just a quick overlook of what's Orion. So this is a pharmaceutical company placed in Finland, and here's some numbers. Maybe something to highlight here. The R&D budget or investments per year is more than 100 million. I think Juha mentioned a couple of hours ago in the introductory that the total R&D investments in Finland, I think that was 480 million. So whatever it is, it's something like a fifth of that. So it's significant. Established more than 100 years ago, some 3,000 people working on. And what we do, so we make medicine for humans, obviously. We have our own proprietary product research, and we're focusing on oncology and pain. So cancer and pain medicine. And that's where I'm located. I'm at the medicinal chemistry, looking for new pharmaceuticals. And in addition to this, we also have chemists doing process chemistry for us. So the whole fermion side is our process side, scaling up all our active pharmaceutical ingredients and producing all our own proprietary products for the market. And both of the medicinal chemistry as well as the fermion we're both located in Espoo, in the capital region of Finland. All right, so just want to give you a little bit of overview. What does it require to do medicinal chemistry? What are the requirements for the chemistry that we need in this business? So this is a very sort of simplified view of how this works. But in principle, when we start to look for a drug candidate, we typically have some sort of idea of a biological target. Let's say a protein, for example, and we might have some idea of how it looks like, how it's forming, what kind of pocket it has that we want to have an interaction with, and we might even have an idea of a ligand that is already binding to it. And put those together, we know where we want to bind, we know already what binds to that pocket. We can start to design pharmacologically better molecules. So that's our first step, design. And the design is purely on the properties of the molecule. And everything we design, obviously, it needs to be something that hasn't been done before, right? It doesn't make sense to make a molecule that's been done before, because then there's no way we can make that into a product. So we design a new molecule, something that has never been done before, and then that gets given to me. Make that. That's my job in simple terms. And all that matters is that I make it quickly. It doesn't have to be nice, and it doesn't have to be fancy, but it has to be fast. Once I make it, it gets sent to testing. Finally, we analyze the results, and then we come back to the design again. Design a new molecule, once again, solely on the properties. Doesn't matter how easy or difficult it is to make. Once we design a molecule that we think would be the best next thing to make, then it comes to my table again, make that one now. And you can see, so the only thing that really matters in this box is how fast is it. How fast is this? Nothing else really matters. You just have to get it done. All right, so that's the sort of the background where we at. So quite different from academic research. Gives you sort of different pressure on the chemistry that needs to be done. All right, so what does it look like? We can, for example, imagine we have a case that we run a library of compounds based on this sort of CN coupling, for example, given here. Could be a Buchwald coupling or Ullmann coupling. And we want to have these aromatic anilines of some sort coupled to, for example, heterocycles. And each and every coupling we make, every single one of them will be a new reaction. And you can imagine that there are, for each of every of those reactions, thousands of possible reaction conditions. And probably if you give a never been done before coupling to, say, 5 different chemists, based on their different backgrounds, their preferences, based on how well they can utilize search engines, they will probably all pick 5 different conditions. It's quite likely, especially if it's a coupling like this. It's pretty likely that they will not find the same conditions. And then they all 5 run the reaction, and since they all pick different conditions, you get varied results. Most of them, if you're lucky, work somewhat, but then in some cases you have reactions that basically fail completely. You don't get anything. And now for the medchem sense, it doesn't really matter whether you get say 90% yield, as here the chemist A as an example, or 50%, for example, as chemist B. These are both successful. You know, given you started with enough time material, both of those two got enough. You have enough product, you can go forward. What's not great is that if you end up with the situation with chemist D, you don't get product and you lost time, then you have to go back and redo the reaction. And this is something we want to avoid. What we've sort of envisioned here is that if we had some sort of machine learning or AI or whatever we want to call it, tool that would help our chemists to pick conditions that will work. Now, again, it's important, the conditions don't have to be perfect. We can, for example, in the case of chemist A, if the chemist A takes the conditions that the computer gives him in this now imaginary example, he will actually get less yield than he would have on his own intuition. That doesn't matter. Again, E as well gets a little bit less. That doesn't matter. What's important, that now chemist D succeeds, and they actually all get unified, predictable results. And also important that we can now refeed the results back to the AI and tell it that this worked. So this was good. And that will continuously improve our predictions. Right, sounds great, but this is actually extremely difficult task to do. And yes, less variance, that's very important. So what do you actually need to get this done? Well, first of all, to tell a computer what a reaction is, we need to put this to some sort of numerical form. The output is, well, easier, let's say. Selectivity, especially, that's great. For example, especially say energy selectivity, that is directly correlated to the energy differences of the two competing transition states. So that's perfect. That's very clean data and unaffected by anything outside of the reaction conditions. Yield is a little bit more complicated, especially if it's isolated yield. You can imagine that the yield is affected by, of course, the reaction conditions, but also the way the workup is done, the way it's isolated, the way it's purified, how clean or how dry the actual isolated product is. All of that affects what we report as isolated yield. But anyway, yield is still a number, so that's fine. We can put that as an output. But then on the input side, we need a way to describe everything that goes into the reaction. All our reagents, all our reactants, all the catalysts, all the conditions, somehow in a systematic numerical way. And that's not an easy task. There are obviously been developed many ways to do this. For example, for sterics, you can use this sterimol. That's one way to describe quite efficiently, perfectly the spherical environment of a given atom. Also, you can calculate IR vibrations. You can calculate NBO charges of given atoms. The problem is that both of these require rather heavy computational tools and DFT calculations and conformational analysis, especially for the sterics. But it can be done. What's sort of a little bit easier, a lighter at least computationally, but then perhaps doesn't contain as much physical information is this typical sort of QSAR type parameter, such as just simply how many hydrogen bond acceptors does your molecule have? Just a number. How many hydrogen bond donors do you have? What's the log P? How many rotatable bonds do you have? What's the molecular weight? These are numbers that can be generated simply from a ChemDraw file very easily. And you can easily imagine that this is something you can generate thousands of very easily for each and every reactant. The same goes for the catalysts as well. The same sort of parameters can be calculated for them. Conditions, especially solvent, requires a bit more work. So there are ways to describe solvents in a specific way. You could also use the same parameterization as shown above for the reactant, also for the solvents. Why not? It's possible. But something to be considered as well. Temperature, time, concentration, those are all numbers, so that's fine. So in the end, what you end up is a whole variety of different numerical descriptors, and put this all together, and you have a set of numbers that describe a reaction. And you can imagine a reaction with a couple of reactants, a catalyst, base, a solvent, and all of that. You end up with thousands, easily thousands of parameters. So what has to be done next? Well, it's fairly worked out mathematical problem. You have your predicted output, and then you have a set of parameters, and you have a script that simply goes through the parameters and finds the ones that have the most impact to your output. And you put X, Y, Z multipliers in front of them, and you fit a line. That's, to put this very simply. Here's a couple of reviews on this field. So it's fairly well established. And this works really well when you have a isolated chemical environment. So if you want to model, say, if you were doing a process chemistry and you have a set goal, this is exactly the molecule you want to make. You have an isolated sort of chemical space that this is the reaction I want to make. This is a way that can be used to optimize a reaction very efficiently. Similarly, for example, photosynthesis is an environment where this could work very well. And we just saw earlier in Petri's presentation example where something like this was actually, I think, also attempted. All right, but this is for an isolated example. The thing is that what we're working with is something like this. So what this cool sphere here is representing, this is a part of our chemical space at Orion, mapped using this EuChemS reaction tool. So what this does is you feed this machine learning script everything you have in your lab box, and it reads the reaction as a sentence, and it starts to look for similarities, and it starts to pair them together. It starts to find sort of families and superfamilies and clusters, and it starts to form this tree of a kind. So every little dot here is a single reaction, and once you have dots in a branch like that, it means that those are related reactions, right? So I can't, of course, show you what exactly is in there, but we could imagine, for example, that here on the side of this space, we have all our nucleophilic aromatic substitutions. So this little branch contains all our SNAR reactions with different nucleophiles and different aromatics. And you can imagine that this isolated space could well be described using the way I just showed you earlier. So simply isolating the problem, now we can make a predictive tool to work on this space. Similarly, we can imagine that on the other side of the map, we have all our Suzuki reactions, for example. And here we have a lot more space, a lot more varied structures. But again, if we simplify it enough, I sort of limit the space enough, we could again run the similar predictions in this space. But that's not really what we're after here, right? This is the state of the art that can be already done. What we would like to do is to model all of this. And well, how would you do that? You can imagine, for example, the SNR reaction and the Suzuki reaction. You will have overlapping substrates in those reaction classes, but the parameters to describe how those reactions work, most likely you cannot combine them, right? So whatever is good for Suzuki is not necessarily good for an SNR reaction, right? So you need to do something else. And one way that we're working on, we're thinking of, that you could, let's say you give this script a new reaction. Let's imagine this is an unknown reaction, It's not, but let's imagine it was. First thing we need to do, we can locate it. So where would this reaction be? And we can find the chemical space where this belongs. And you can imagine this could be done quite quickly. This is a fairly simple script. Next thing is we can isolate now. So we know where it is. We can quite easily sort of imagine that we could now run a similarity search of reactions that are, for example, 90% similarity to the one that you just given, for example. Once you've done that, well, then you have an isolated space again. And looking at the isolated space, you could quite easily imagine running classification, for example. So you can now recognize that this is indeed a isotropic reaction again. We have an electron-poor, non-hindered electrophile. Your nucleophile is a heterocycle. It is rather hindered. And based on those descriptors and sort of subclassing it, putting it to the box, you can imagine that you could find that with these substrates, with this sort of subclass of this main class, typically this is the catalyst you want to use. That's the ligand that is typically working the best. That's solvent and so on. And what's important, so what we don't, so what's not enough is just to get this done. We need this to be done very, very quickly. So we needed a go, no-go accuracy. That's sort of the easy thing. So we don't need to know if this will give 80 or 60% yield. Those are both go. But what we need is, will it be 80 or 10% yield? So 10% is a no-go. That's the easy, but the fastness is the difficult thing. So for this to be useful for us, this really needs to be done in seconds. This cannot take minutes or definitely not hours. It has to be seconds. So ideally, the way this is done, that you draw the reaction in your lab book, you plan that this is the reaction you want to do, and by the time you're done drawing it, you already get sort of this pop-up that, well, do you want to try this? That's the goal we're aiming for. We're not there yet, but we are making progress. So, in summary, what we've done is we've had a master worker doing a master thesis on this project, and he's been actually doing the sort of ground up approach. So we've given him an isolated example of an isolated space, running modeling on this space. So that's sort of what's already known to work, and it does indeed work. You can do it. It's been a nickel catalyzed Suzuki that he's been working on, and with success. So great, that's good. What we've also been looking at the same time as he's been working on this is to try to automate every step, try to make it as fast as possible, and try to get every single step of the way somehow automated to remove all the manual labor. So then we could have this piece that we need for the final version of this. Simultaneously, from the ground up, we've been working from top down. So we're working on the mapping of our reaction space, looking at subclassing it, looking for how to divide it into smaller subsets and all that. And well, running into interesting problems. For example, putting a 0 in your lab book for a reaction can happen for many reasons. For example, the reaction actually didn't work and you actually got nothing. That's one reason. One reason is that you actually never did the reaction. You just put 0 and then move on. And that doesn't matter until you give that data to a computer. Because for a computer, 0 is very significant. And that's a big problem that we're working on, how to get around all the zeros. Just to give you an idea of what kind of issues you can run into this. But yeah, slowly making our way from ground up, top down, and trying to meet halfway through. And yeah, finally, want to thank again our generous funding and all our collaborators in this project, and of course, especially Yuri for actually doing all the heavy lifting, and Antti, who's been the master thesis worker on this project. And yeah, thank you all for your kind attention. Hope you are still up. I don't want to stop you from starting a weekend.
Great, Tony. Superb last talk of the day. Well done. Again, presentations open for questions either through Q&A at the bottom of the webinar or by raising your hand. Ari, you're up to ask a question, which you should be able to do as a panelist. Off you go.
Okay. Very much. A very interesting talk. I was wondering about the sort of parallels to what used to be 40 years ago, the QSAR and QSPR study. Going a step further, what about the cases where you really don't know what the reaction is to be sort of combine molecules A and B together, completely new product? In other words, reaction discovery kind of thing. Do you have any means of taking care of the problems like dependence of variables from each other?
Yes, I see what you mean. I think that is a very difficult problem. I've been thinking about it because with this mapping that we have already done, you can map your reactions. Let's go back to have it full picture. But you can similarly look for products from this map, right? So you can draw a structure you want, look for it in the map, and see which disconnections were made, form similar structures like this. Is that what you meant? Right. So we can find how was this disconnected earlier. That's one way, but it's very complicated. It's even this is already complicated, and that's another level of complication.
Yeah. There's still need for the old thing that you just have to. There's one way to know. Just try it.
Yes. So what this needs from the chemist is to draw the disconnection. You still need that.
Yeah.
And and that's still a difficult task for the computer to do. But if this could be done in seconds, then of course the next layer is make the computer generate, 100 different combinations, disconnections for that target.
Good.
Right. That's of course. Yeah.
Yeah. Thanks for your talk. Very nice.
Very good. Cheers, Ari. No other questions coming through. Tony, maybe I could ask you one. I mean, the chemical engineers and those working in process chemistry relied, or at least they do even now, still rely on design of experiment. Yes. Do you think there's much of an overlap between, or potential overlap between what you're proposing to do and design of experiment?
Not really. So the design of experiment, and that is well established, and that is the way to make process. So this is mainly for medchem. So it's a different task. In process, you have time for that one reaction, and the best way to do it, you need the information you get from DOE, not just to find the best conditions, but you also need it for the robustness of the reaction. That's something you want to know. You want to know how sensitive the reaction is for variance. So obviously you want to find the absolute best conditions, but you want to know which way is the hill steepest. That's also relevant information. Whereas in this setting, you just want to get the reaction done in one shot.
Yeah.
Not that it doesn't have to be perfect. You don't have to find the peak. Just want to find a stable, okay condition. That's all it. Well, that's pretty simple. That's all you need.
But that's a lot. So
it's a lot, but it's actually it took us a while to sort of realize that it's actually a very different problem.
Yeah. Okay, I don't see any other questions coming through either in the Q&A or from participants. So maybe we'll leave it there, Tony. And again, thank you for your talk very much.
Thank you.
Unshare your screen. Okay, so there we have highlighted organic chemistry in Finland. I want to thank everyone involved and just some background information on it. We had about 180 people registered as of about 11:30 today, and I was monitoring attendance, all that sort of stuff. And for most of the talks before the coffee break, we were at about 98 to 104. I think 104 was our max, which is a pretty good return I think, on 180 people registered. After the coffee break, I don't know whether people just decided not to come back, but only about 80 attendees from there on in. But look, that's what you get with an online webinar. People dip in and dip out in terms of the talks they want to listen to and all that sort of stuff. But also, depends on the time of day where you are in Europe. I know it's getting close to 6 p.m. in Finland. It's still not yet 4 p.m. in Lisbon or Dublin, but it's probably about 5 p.m. just about everywhere else. So I know how that works. So we started off then with a really good presentation from Juha Siltanen. Good to see you again, Juha. We met only, last week. I'm trying to remember when now. So it was just Friday, Saturday at the Young Investigator Workshop held in Lisbon. So Juha represented Finland very well, I should say, at that meeting. And, gave her an excellent and super informative. Sorry, Ari?
Sorry.
No.
I'll mute myself.
Okay, yep. You gave I would say, an excellent and an informative summary of the current, the history of some organic chemistry coming from Finland, but also the current state of what's happening across Finland, what are the sort of major themes. And certainly it was interesting to find that the chemical industry is worth about 20% of all the individual or industrial products and goods that are exported. So it's quite clearly very important to the economy. So that's good. Then we had 7 superb lectures. We started off with Anu, who's no longer here, but from the PET Center, and again, a great presentation on the design and synthesis and applications of PET radionuclides. So again, very interesting. Petri, 2 Petris, but the first one, Petri Tuhanen, informed us, and we learned a lot about bisphosphonates and some very exciting new work then on DAWX for acid catalyzed reactions. So again, excellent work. Third lecture from Philip, again, the design of boron-containing carriers, modifying sugars mainly, lots of multi-step synthesis, nothing wrong with that. And again, some of the examples were showed up to 100-fold delivery capacities. So that's really impressive work. Then just before the coffee break with Kaisa, who talked a lot about host inclusion complexes and some very detailed NMR investigations of those complexes. Then we all had a tea or a coffee or a comfort break, and we came back to Jan Deska, who's not just bio-inspired, but actually tried to chemo-inspire the system by finding enzymes to do, reactions that they're not normally set up to do. So again, it was a fascinating lecture. Some reactions enantially selective, some not, but it's trying to find in nature the trick that actually, finding the reactions first, and you can worry about other things thereafter. Then we had Petri, who actually I last met in 2007 when he presented at the European Symposium of Organic Chemistry here in Dublin. And again, an excellent talk on designing new organocatalysts, and mainly for forming tertiary stereocenters with methyl groups, and found and designed catalysts that were useful for a range of transformations. And the critical thing is that not only do chemists have to be flexible, actually the catalysts have to be flexible too. So that's pretty impressive. And then last but not least, we had Tony, who gave us a really impressive talk on machine learning, which will optimize synthesis of pharmaceuticals. And not that we don't realize it yet, but machine learning is growing and growing in importance. And I think it's really good for all of the postdocs and PhDs who perhaps zoomed in to this to hear that type of presentation, because when they exit academic life, if they don't have any sort of that machine learning background, they will need to learn it, I think, when they go to industry. So overall, I have to say, a fantastic 3rd edition to the Highlighting Organic Chemistry series across Europe. And that's what I'd like to say on behalf of the Division of Organic Chemistry. Many thanks to Ari and Yuri for putting the program together. All I did was the Eventbrite stuff and set up the webinar. So that's the way it works. So I'll maybe leave the last word to you, Ari, if you want to say anything, or Yuri, on behalf of Organic Chemistry in Finland.
I would thank everybody, all the participants, all the speakers, and everybody who here listening to this talk. More people will see them later on in the YouTube. But I think it's appropriate if Yuri will say the last word, because he was the guy who also. Thank you, Yuri, also for your great work.
Yuri, pass over to you.
Thank you. Yes, it has been excellent day. Actually, I wasn't waiting this good participation, but it was very nice to see that many people, and also which is interesting, I checked once and then participants. So I guess we have more than 120 different persons visiting because they changed a lot. So
yeah,
it's very nice. And also I saw a couple of questions already in the LinkedIn about missing this. So I think the YouTube will be very nice.
Yeah, the only bad thing about YouTube is it takes my computer about an hour and a half to decompress all of this before I can go home. Like that's life anyway. So,
yeah,
you know, we learned a lot about organic chemistry in Finland, and the bizarre thing and the experiences we've had both in Italy and Ireland is that the people in the countries being highlighted, they actually learned a lot about what's going on in their own country as well. So there's a positive to that also. So, you know, it's been a great day. Thanks again to all involved, to all the speakers, to everybody who attended. Not quite sure where the next one is going to be for Q4. It may well be Portugal. We'll just wait and see on that. But for the coming year, we have a range of countries, Catalonia and Societies Catalonia, possibly Spain, the Czech Republic, UK. So there's a bunch of people aiming to do it next year. And that's the whole thing. We just roll through 4 every year, 1 every 3 months. And by the end of that, I think we'll have a great repository of what's happening in organic chemistry across Europe. So have a nice weekend, everybody. Thank you very much for participating. Bye-bye.
And before you stop, thank you. I think this has been a fantastic exercise and from you organizing. Thank you very much.
No, you're very welcome. You're very welcome. Okay, thanks again.